I suggest we make a start in the interest of time. We've got a busy and fun-filled morning for you, so let's get the show started. Welcome, first of all, to everyone. It's great to see many familiar faces and names on the call, and people who've been involved in the project throughout. It's also good to see people who we haven't met before and people who've been perhaps interested to hear what's been going on in this space. So a warm welcome to those people, and we hope that you'll find the morning interesting and perhaps some more new material for those people. It's a grey, almost autumnal day here in Cambridge this morning, but it is indeed spring 2023. And that means that we've recently finished in December the official funding period for the multi-omics Cohorts Consortium. So today is a chance to celebrate. It's a chance to share the successes. It's a chance to provide insights into the challenges we faced and how we've tried to overcome them. So it's a chance to reflect and look backwards in one sense, but it's also a chance to tell you all about what's been going on, and also think with a view to the future as well. So although the specific funding for this project is finished, as many of you on the call will know, HDR UK is about to start its second 5-year cycle. And so a lot of the infrastructure and the science that you've heard about today will hopefully continue as part of the new molecules to e-health records theme in the second 5-year cycle. So it's also a chance for us, I think, to take stock, to see what worked well, to see what kind of things we didn't manage to cover in the first 2 years that we want to pursue. We're doing this in a hybrid format, of course, so you can join from the comfort of your offices or homes. It's a shame not to be in the same room as you all, to be able to share a cup of coffee or a lunch together, but we hope that the convenience and comfort will outweigh the lack of close contact. Loma's organised a busy agenda for us. We're aiming to wrap up about 12:30. We will have a break halfway through, because sitting staring at a screen for several hours can be draining, even from the comfort of your home or office. And to start with, I just want to perhaps introduce the project to people who don't know so much about it, give a bit of background and a bit of introduction to what's coming over the next few hours. Although we're all sitting behind computer screens, I hope that people will engage and post questions. You can use the chat function at the bottom of the Zoom if you want to make any comments or questions, and we have tried to encourage speakers to leave 5 minutes at the end of their slots for some audience questions. So what is the HDR UK Multi-Omics Cohorts Consortium? It was as the slide says here, this was what was called a national implementation project. This was an effort to build beyond the regional hubs that HDR UK originally funded, to try and do some nationally joined up work. So alongside other projects that focused on multi-morbidity or machine learning, we decided to focus on multi-omics. And what does that mean? Well, this slide hopefully illustrates the challenge that we set ourselves, the opportunity that we saw, which was really to try and, there's been lots of success in the past couple of decades linking the human genome and genetic variation with disease outcomes. But we wanted to see now there was lots of things that one can now measure in the blood beyond the genome. We can measure RNA instead of DNA. We can measure proteins and lipids and metabolites. The technology to capture these things at great scale had really matured. So we wanted to see to what extent could we really bridge that gap? Could we really understand more about causes of disease in particular by interrogating this layer of biology? And to do that, we wanted to capitalize on the rich resources that the UK had built up over the past couple of decades, particularly in population cohorts. And you see many of them down in the map on the bottom left, which had recruited huge numbers of participants from around the country. All of these cohorts had some kind of genetic data, so we could interrogate the human genome. They all had linkages with electronic health records and NHS databases, so we could look at which people had which conditions and diseases, And they all had some kinds of multi-omics data, proteins, metabolites, transcriptomics, etc. And so the real goal of this project was to see, could we build on this existing data? Could we bring it together and form a collaborative consortium that would help to generate insights that perhaps couldn't be gained from individual studies or individual platforms? And you see on the right some of the questions that we set out to address at the start, and hopefully during the course of the day with some of the projects and examples that people highlight, you'll see some evidence of those. How did we organize ourselves? Well, we organized ourselves into kind of 4 different groups, and you'll hear from each of these groups and work packages today. Underpinning at the bottom, of course, was the population cohorts and the data. And of course, at the top, importantly, was connectivity with with stakeholders, particularly with HDR UK and its innovation gateway. Importantly, with public and patient representatives, and of course, cross-linkages with other HDR UK projects. In terms of what we're hearing about today, well, we're going to hear actually about the challenges and successes in engaging with public and the patients from Amanda and Sarah in Cambridge, who led our PPIE work, and from Chris Cunningham and Jenny Bovard, who are our representatives on our steering committee. So we're going to kick off with that. I'm going to tell you a little bit about what I see as a project lead in terms of the successes and the challenges, and give some examples. We're going to hear from Emily Jefferson and from Chris Orton, who co-led the first work package, thinking about how could we create IT and informatics solutions to enable us to analyze these data, and what needs to be done to make this truly usable, findable, and workable resource. Joanna Suzlaki is going to tell you about some of the work that we led in the metabolomics space. And there was a parallel work package on proteomics. You're going to hear from Arun Hingarani and Mike Pietzner, who are closely leading that one. And finally, we wanted to think not just about individual types of data, but about how we might integrate across different data types in a work package led by Chris Yao and Mike Inouye. And Mike's going to tell you a bit about some of the work on that today. I'm conscious that these kind of things, we often run out of time, and the bit that we try and do at the end, where we say thank you to everybody, gets lost and brushed under the carpet. So I'd like to really put up in lights some of the key groups and key people who've made this project such a success, particularly the partners in crime, the work package co-leads and teams, many of whose names I've just told you about, who you'll hear from during the course of the day. They really drove the project forward and led the pieces of work in each area. So a huge thanks to them, because the science wouldn't have emerged without them. You'll hear in a minute from the PPIE team. So thank you particularly to Amanda, Sarah, Jenny, and Chris, who've kind of spearheaded all of those activities and really helped us navigate what can be done in a project like this, and to understand how to hopefully during the course of the project improve in our engagement and inclusion activities. There's a big team behind the scenes, particularly here in Cambridge and HDR UK, who've helped to do all the boring bits, sending out the agendas, setting up the meetings, making sure the budget is being spent, coordinating across all the different centres. So particularly thanks to Richard, Amy, Loma, and the operational team. You'll hear from a few people today, but of course the work that's being done is really thanks to all the people involved in all the different cohorts, the people who lead those resources, and of course all the research scientists, the PhD students, the postdocs who actually did all the data analysis and led the work. So a huge thanks to those people, because of course none of this would have happened without them. And finally, thank you to the funders at HDR UK and the central team, who've been a huge support. They've engaged with the project throughout. They've helped steer us on our way. They've given us critical feedback at useful times to help make sure that we're pointing in the right direction, and really enabled us to do national science rather than local or regional science. So with that, with a big thank you to everybody, I'm going to stop, and I'm going to hand over to, I think, Sarah and Amanda and Chris, who are going to tell you about our public and patient involvement work.
Brilliant. Thanks, Adam. So I assume I'm okay to share a screen. Got some slides. Can everyone see slides?
Yeah, can see those.
Lovely. Some good thumbs up. Great. So thank you very much for giving us the opportunity to share some of our findings and our reflections from the PPI work in this project. And I think a lot of projects actually don't take the time to reflect what the outcomes were. And Chris and Jenny and Sarah and I at a really interesting meeting a couple of weeks ago to really have a think about, how it all came together and which bits worked and which bits didn't, and what we really wanted to share with you today. So as Adam has said, our PPI team for this work was kind of made up of a 4 team of the 4 of us, our partners in crime, with our 2 key public contributors, Chris and Jenny. And such is the commitment of Chris and Jenny to this project that Jenny is dialing in today from Australia, from all that way in the evening, and has come. So a huge thanks to both of them for really sticking it out with us and helping us to consider some of these really deep questions, because it was a really complex project. Also, huge thanks to Sarah. It was really helpful to have 2 of us, I think, working on this project, able to bounce questions back and forth and kind of find a way through what was otherwise, as we keep saying, a really quite a complex area. I work on a lot of health data science projects for patient and public involvement, and the complexity of the omics really added an extra dimension to this. So how did we put it together? Kind of the general way that we structured our patient public involvement for this project. At the beginning, we worked mainly with 1 public contributor. So Chris joined early and worked on a one-to-one basis quite regularly with Sarah. And both of our public contributors attended the different types of meetings. So many of you who have been involved in this project over time know that we have the regular multi-omics meetings that take place in Cambridge, which are kind of operational and sort of the decision-making type meetings. And the public contributors also attended the regular multi-omics steering committees that were a little bit bigger in nature. So they got to be involved in all types of discussion and hearing results and in the decision-making type processes. And then as a way to kind of complement that or supplement that or build on that, the public contributors often would meet with Sarah and sometimes Adam after meetings to kind of go through what was discussed, because sometimes there are questions that either you don't think of when you're in the middle of the meeting, or just things that you need to clarify at a later date. Now, obviously, it would be much more interesting to hear from one of our public contributors themselves about their reflections. So at this point, I'm going to hand over to Chris, who has very bravely volunteered to share some of his reflections of being involved in this project. So Chris, over to you.
Thanks very much, Amanda, Adam, everyone. So yes, I'll talk a little bit about the successes and challenges I saw as a public contributor, and also perhaps touch on some of the wider reflections that we as public contributors had, and which I mentioned in some of the later slides, I think. So just to say, my professional background is in international child rights development, and my sort of previous experience of genetic related work amounts to practically 0, so you can see why I'm such a good fit. Anyway, I think Amanda has said this. I joined at the outset and sort of stayed pretty much throughout. Was very happy to be joined by Jenny, both now and as public contributor a few months after I began. Her background is in, well, much more relevant areas than mine, nursing and related research activities, I think. And we were so well supported by Sarah and Amanda, particularly as the public involvement and engagement coordinators, the professional ones, and of course by Adam and all the other multi-omicists, if that's a word, involved. So on the challenges, yes, indeed, as they say, there were a few. So I suppose the starting point was the unknown scientific territory, certainly for me, a plethora of unfamiliar terms, some very difficult and in some cases undecipherable, I found, acronyms, and of course all the different omic strands that we had to contend with. And indeed, I found myself, particularly at the early stage, sometimes wondering if people like Adam occasionally threw in a few sort of fictitious omics branches just to catch me out. And if you did, Adam, it worked. But moving on to another perhaps sort of, kind of harder challenge to crack. It's the role definition of a public contributor in an area as specialized and data-driven as multi-omics certainly is. And, just by reference, I'll compare to what is perhaps a much more, well, hope is a much more obvious need to gain patients' insights into a health service treatment or service that they've received or taken part in. And I think, probably it's fair to say to this day, we haven't really sort of cracked what specifically that public contributor role is in an area like multi-omics. And finally, on the challenges, I just want to talk about the external workshops that happened. Of course, the scientists were eager to share some of their developments and discoveries with other external scientists as the project developed, and very welcome. They wanted other public representatives, including ourselves, to take part. At the best of times, a mixed workshop like that is going to be difficult, but because of all the challenges, the specialism of an area like multi-omics, I think it was a particularly difficult balancing act in this case. I won't say more because as a public contributor, I wasn't particularly involved in the organizing of the workshops, but I'll leave it to the scientists and perhaps Amanda if they want to say more about that. So then moving quickly on to the successes, and there were plenty of them, and probably certainly more than I've got time to address now. So I think it has to start with the very warm welcome we received from the scientists. And as I think has already been said, both Ginny and I think, felt part of the team from the outset. So thank you very much for that. And coupled with that were these sort of meetings with Sarah and Adam that we had during the course of the project to help us unwrap, to help us better understand some of the difficult terms and concepts that we were meeting along the way. And through that sort of combined warm welcome and all the explanations, I think that really enabled both Jenny and I to feel more able to take part, to ask questions, to raise issues, and from that we indeed saw a direct impact within the meetings themselves, within the consortium meetings themselves, on improved clarity of scientific communications as far as we were concerned. So, for example, the scientists took time to slow down, to explain some of the acronyms, and so on and so forth. And then on to the external workshops. Well, for us, that was a great boon that we were invited to take part. It meant we could widen our messages on issues of terminology and clearer public communications to a whole host of scientists, academics, clinicians, managers in often quite influential positions themselves. And, we certainly tried to do that, I think. And even more specifically, we were invited on to other meetings. So, for example, Chris Yao, who was leading some training work with postdoc students at HDR UK, wanted us to work with some of his PhD students to help train them in better public communication practice in early stages of their career development and as they embarked on their omics and related research. So, for example, we would hear some of their presentations about their research and comment on what we'd found understandable or not so understandable, and make suggestions as to how they might think more and improve that. And that was great. And just one final area, because I know time is short, I will mention polygenic risk scores, PRS. PRS is, well, I would say it's a way of using your own your DNA information to help predict your future health outcomes. And so it's a potentially huge public interest. That work was led by Mike Inouye and others, and I'm sure Mike and others will lead me out afterwards and explain to me why I certainly shouldn't attempt a definition of PRS in public ever again. But the important thing is it was a really collaborative effort between the scientists, between the professional public involvement coordinators, Sarah and Amanda, and Jenny and myself, and other public and patient representatives to try to unpack PRS so that it might be more approachable by a lay audience, if you like. And again, I'm not going to say more about that, because it was Sarah, I think in particular, who took this work forward in the public domain through some really inspiring work with young people in Cambridge. And, you know, maybe she will have a chance to say a little bit more about that. So I think I better stop now. I can see Adam looking at his watch. But that's it from me. Thank you all. Thank you very much for listening. As Adam says, please put any questions in the chat box, and I'll hand back to Amanda for a few words more.
Brilliant. Thanks for those reflections, Chris. We've just got 1 or 2 slides more from Sarah and the team and I, and then we'll go over to questions. So, as I said, the chance to reflect was really helpful for all of us, really. We had a bit of a reminisce over whether or not it might have been helpful to have one single dedicated PPI professional for this project. I mean, Sarah and I both believe that the ability for us to bring in our other projects and crucially to take this learning out into kind of our wider work was really helpful. But because it was so complex, I think both of us sometimes felt like if we'd had all of our time to get our heads around it, that might have worked better. And then also, as with many projects that took place in this time, because it was mostly online, it did mean that our public contributors and ourselves didn't get the chance to interact just over coffee or over food or anything like that, those few short conversations at different events that we otherwise would have. And I think we all felt the loss of that, although the online worked really, really well. As Chris alluded to, sometimes it's not always clear exactly what purpose public contributors have in meetings like this. And we actually had really interesting discussions on how sometimes it's not important to know in advance. The fact that it worked anyway, and that we all worked together to make it work, to make sure that there was usefulness in it, was really beneficial in the end. And then, as Chris said, this project has gone on to spawn many other public involvement activities and opportunities, and has certainly informed my practice and what I teach researchers. Now, Chris has gone into some of these. I think these slides are going to be made available, so I'm going to kind of skip over this with you being able to read this later. But we really wanted to sum this up by handing it over in some ways back to you. We would love to know what you all, as scientists, as clinicians, as people leading work in this health data space, if you also had learnings that you learned from working with Chris and Jenny, that you learned from working with Sarah and I, we would love to be able to hear about them so that we can write them up and include them in our project, and crucially to share them with everyone else. So I'm going to hand back to you, Adam. I can stop sharing, and then we can take questions if there are any.
Wonderful. Thanks very much, Amanda, Sarah, Chris, and thank you for joining us from Australia, Jenny. I don't know if you wanted to add anything, your reflections.
I'm still slightly jet lagged, I think. I only arrived Tuesday.
Bless.
Anyway, just to say, yes, I would agree with everything. I contributed to that. As people know, missed an opportunity, but also to say thank you, like Chris, to the whole team, because this was, I found this really challenging, and being made to feel that a contribution was welcome by really you very, very clever and advanced scientists was a really good experience. And I feel I've learned quite a lot. And I feel I constantly drop omics into conversations. because it baffles even more people, and it sounds as if I've got very, very learned, and it makes a great impression until they ask me to explain it in detail. Then I tell them to look at the experts, and I can tell them, give them dates, people to contact. But anyway, it's been a really enjoyable experience, and you've all been really kind and helpful, and I've like Chris, I've found it very rewarding. And I wasn't sure I'd be able to join you, but my nephew and his wife, she needed to get to bed early, so I got back to my hotel room early.
Oh, great that you could, and thank you particularly to Jenny and Chris for spearheading the involvement from the public representatives, and just to say, I think from on behalf of the scientists on the project, your input was invaluable. We certainly learned a lot, and I'm pleased to see Amanda's keen to collect learnings. I think you challenged us. You gave us different opinions to maybe what we might have expected, which was, of course, a key point. And if I take one thing away, it's that whatever piece of lay text I write, it can always be improved. It can always be made clearer. And however good I think it is and interpretable it is, it needs that input from public representatives to make it really understandable. So thank you very much. I can see we've got some hands up, which is great. So I'm going to turn to Morris first, if you wanted to ask your question.
Hi there, I'm a lay representative, and I think I'm on of HDR UK. I think I'm a member of the PPI group, if there is one, and I think what you've done on the PPI in relation to omics and to data is very important, and I think we should have a meeting at a central meeting so we can discuss this in more detail, and maybe with other projects about how to tackle data and omics. Now, my question is, what happens at the end? How is the PPI going to be looking at the results? Will there be lay summaries? How to implement? All those sort of things get sort of overlooked in ending of projects like New Year's Eve. You're rushing and so forth. So could you tell us something about how Chris and others, and I think there should have been probably more representatives. 2 is probably not enough. Whether you've got to sit on all the meetings, I don't know. And if you've got Zoom, it's more accessible. But I think there is always an under sort of scrutiny of what happens at the end. And are the representatives going to be joining in to write up? Are they going to be mentioning papers and so on? So could we give us some indication there? And also could we have it at, take what you've found to the central so we could discuss it in greater detail and have, how to go forward. So I welcome what you've done so far.
Yeah, no, thank you. And actually, I rushed through my last slide a little bit at the end because one of the things that we're really excited about is the ability to share these findings. We think they're really important to share on a wider scale because it has been so challenging. Sarah and Jenny and Chris and I are planning to write up some lay summaries looking at both some of the findings and some of the sort of successes and failures, as it were, of our PPI activities. We will certainly be sharing these slides with anyone who will sit still enough to read them. And we also want to make these reflections available to share, because we think for future HDR UK projects, for future health data projects, and certainly for future omics projects, that these learnings are really crucial. We spent 2.5 years working on them ourselves. It would be a shame for any new team to start on them again. In terms of the more public contributors, yes, I think there is never too many that you can have. And I think one of the challenges, though, of the omics project and the complexity is that it made it much harder to do kind of like one-off events to dip into people, because you needed to understand a little bit of the background. There is so much, a lot of people found it quite scary to even engage with, which is partly why it works so well to have our ongoing relationships with our public contributors. We did do a few kind of wider activities. We had, there were some very brave people who sat through a public engagement activity with me about what omics were, as I attempted to explain it, using a very rainbow slide in an attempt to distract. But we did do a few activities like that, which also fed into our learnings. But I think it is one of the things that would be useful to think about going forward as well. How do we make this kind of event more inclusive when it's hard to make health data PPI inclusive in the first place, and even harder with a topic that's like this, which shouldn't mean that we shouldn't try. Of course we should try, but I think hopefully some of the learnings from this project will make that easier going forward.
So could I just ask you to take forward to central office, you know, the what you found, so they could set something up?
Yeah.
And maybe when you have a lay summary of your findings, that should go out to a wider audience to comment, because if, for instance, if I can't understand it, you haven't done the job.
No, indeed.
And how it's going to feed into the health system, which is very important. So, but I congratulate you it's a hard job. It's a really hard job, but we have to learn about future, because you're doing things that data people think, oh, it's just data. You know, the public have got, so what? I'm not saying the so what bit, but that's how, you know, so I congratulate you what you've done and take it forward. Well done.
Thanks, Maurice. Some great suggestions in there, and yeah, capturing the learning is really going to be really important, isn't it, Amanda? Anthony, I can see you've got your hand up as well, so why don't we come to you, please?
Yeah, sorry. So I'm really late to the party because I just sort of joined HDR I saw this event come up, and you know, I've got an interest. So I got a professional interest as I'm a biomedical scientist by degree, worked in medical diagnostics, had a postgraduate diploma in genetics. And as I said in the chat, I'd find some of this a bit mind-blowing, even with the TARDIS database. So I'm always running away from dialects or whatever. So, so I put in the chat, would it be in future projects, would it be a good idea to have an upfront, let's make this difficult subject or mind-blowing subject easier to understand? So that was one in the chat, but I've also thought of other things. So so I'm involved with PPIE because I don't work anymore, but I'm giving back that way. So so there's multiple projects I'm involved with. One of them is education. So I think that it'd be really powerful if you could get PPIE people in the room with students to sort of explain to them why they're important and why their lived experience is important. Because I'm doing that at King's College with an MSc in psychosis. And it's like a pretty groundbreaking scenario, but I think it's something that I'm sort of evangelizing now, because I think it's really important. So I suppose this is to Adam or Amanda. You know, who would want to answer that? I mean, is that something you're thinking about going forward? Because obviously, if you want more people involved, you need more people to understand and sort of understand why they should be involved. Do you know what I mean?
Yeah. Do you want to take that, Amanda?
Yeah, no, that's fine. So in terms of training our upcoming students in patient and public involvement, certainly in Cambridge to a large extent we already do, and I know because I'm the person who does the training. So on the population health sciences MPhil, I do a section each time. I usually work with some of the med school students and also with the Department of Public Health. Quite frankly, anyone who will sit and listen to me talk about the importance of PPI will certainly get a serve. And in fact, I need to run off in a few minutes because I'm doing PPI training today. I also think there is probably definitely space for a specialized course that works with particularly data science. Not that they need any sort of extra work as it were, but because the topic is so complex. So you can't just start with a bit of a spiel about why PPI is important. It also has to have some public engagement work that goes with it. As Chris mentioned in his talk, even within this project and with aspects of this project, we had the opportunity to work with some students and to develop some learnings in that space. And Sarah's also gone on to do some really amazing theatre work with sort of high school students and things like that. So coming back to Maurice's point about writing everything up, perhaps one of the things we could focus on is the learnings for PPI training and working with students.
And just as an aside, would you try and do future projects in a more co-productive manner? Because that's also a buzzword. And I'm also on a co-production course at the moment. So is that something that you would look at? I know the co-production is a big buzzword at the moment, but it really is quite powerful. And if you're going to have PPIE, that sort of dials into all that. So it's like getting viewpoints from the very beginning.
Yeah, no, I think, and I'm mindful of the time, so I'll try and answer this quickly. But I think that is one of the things that in our learnings to take back to HDOK that we would like to see them allow time and funding for in the start, because it does mean that you need to be able to slow things down. And I think one of the other things that all of this work from these type of projects will give us are people who are already skilled and trained and able to join that co-production from the beginning, potentially in future projects, and give us the opportunity to bring even further people in as mentors and things like that.
Yeah, cool.
Brilliant. Absolutely, thanks, Amanda. Definitely, I think this project, from what we've heard from Chris and Amanda, we thought about some of this afterwards, after the start of the project, and actually designing it from the start. And for example, what are the roles of the public representatives is really important, and as Chris has identified on behalf of the team, was a challenge that we evolved as we went along and maybe didn't ever find an optimal solution to. So that's really important learning to capture because of the science. We'll carry on in the next 5 years of HDR UK. How do we make sure that we're doing better at that? So thank you to all the public contributors. Feel free to post more questions in the chat. In the interest of time, I'm going to move us on, but I will also put Amanda's email in the chat, I think, given that she's keen to collect some of the learnings from the project so that people can reach out to her afterwards and produce some of the reports and the stories and the summaries that we've just talked about. So thank you to everyone involved in the PPIE effort. It's been a really fruitful and a journey we've really learned from over the last 2.5 years. So next, I'm just going to give you some reflections and some examples from the project and what I see as overall as the successes and challenges before we then dive into some of the deeper work packages. I'll start with the challenges because there were many, and this is maybe a summary of some of them, and you can maybe see how well you think we've done by the end of this morning's Insight Day at addressing them. Perhaps firstly and most obviously, we kicked this project off in April 2020. We'd all just been sent home. We were all still trying to work out how to use Zoom and things. So to answer, I think Maurice's question earlier asked about the meetings. The vast majority of the project and the meetings have been entirely virtual, which has in some ways been a challenge. But of course, the idea of this project was to bring people together nationally. And actually, they're using the technology has really allowed people from all corners of the UK and indeed beyond. We have people here today, I can see, from Australia, from Estonia, from other parts as well. So there have been some benefits from that. We've just heard about the challenges. How do we work out how best to optimally involve public and patients? We've learned a lot. We've done some very valuable work, but that was clearly a challenge for us. We hadn't really done that before. We'll hear next about some of the challenges in kind of an informatics infrastructure. So how do we bring the data together to analyze, which is something that is evolving very quickly and that HDR UK is really helping to drive forward. And one of the other challenges we had that particularly the peer reviewers of our funding proposal were concerned about is how could you had all these different studies that had perhaps tried to measure proteomics or metabolomics, but using different platforms, different companies who'd made these different kits for measuring these things. Was it really going to be feasible to bring all these together? So I'll give you just a few examples in the remaining time to hopefully illustrate how we rose to some of those challenges. Well, firstly, perhaps COVID was not only a challenge, but an opportunity. So as we started this project, perhaps some of the diseases we thought about tackling first got sidelined. As we said, well, what can the community do to help with understanding this new devastating disease? Various projects then that people said, well, what could we look at in relation to COVID? And here what we did is we took data from multiple of the cohorts, particularly on the layers of proteins and of RNA or transcriptomics. And we tried to say, as studies started to emerge, looking at genetic variation and people who got more severe COVID outcomes, we said, if we put the proteins and the transcriptomics in the middle, as I exemplified earlier on that kind of horizontal flow diagram, can we pinpoint some of the key proteins and some of the key genes that might be involved in COVID-19 to help understand what some of the biological mechanisms are and therefore what some of the treatments might be. And you see here just some of the names of the genes and proteins that we found. So one of the ones we might have expected to find was this one right on the right called ACE2, and that's the receptor protein for the COVID virus. So that kind of made sense to us and showed our method was working. But we also, perhaps one of the more important ones we found was this one called IFNAR2, which pinpointed the role of interferons in COVID-19. And of course, a lot of the other work coming out at the time and subsequently has pointed to that as an important pathway, which is now being successfully targeted with drugs to help people who've caught COVID. So there were some successes of that quick pivot, and that's work that we published in 2021. Addressing the question of whether or not we could integrate across the studies, but also across the assay platforms. Here's an example of nice work led by colleagues at the MRC epidemiology unit. Here in the figure, you see lots of different metabolites that you can measure in the blood. And the colors here, as you see from the key, they relate to different studies. And in the brackets here, you see different platforms that we use. So there were 3 different platforms that we used from a company called Biocrates, a company called Metabolon, and a company called Nightingale. And although some of the metabolites are only measured on 1 or 2 platforms, you can see at the top here, some of the metabolites were measured on many platforms. And that allowed us to get very big sample sizes, particularly for some of these amino acids, whereby combining across the studies and the platforms, we could get more than 80,000 people who had genetic data and these amino acids measured. And of course, one concern is, are they really measuring the same thing? Do they give you the same answer? Well, reassuringly, when we looked at what predicted heterogeneity or differences between the results, it didn't seem to be so much about the assay platform or about details of the study, but really just about how strong the signal was. So the very well-known examples that have been found in much smaller studies tended to have more divergent results. So we were kind of satisfied that whichever platform you are using, you were getting kind of similar answers, which was really useful for then pushing forward the rest of the work in this space. That's not always the case, though. When we looked at some of the proteomic platforms, for example, on the left here is a plot looking at genetic associations with one platform called Olink on the vertical axis and one platform called SomaScan on the horizontal axis. And although there were lots that lay along the diagonal line here that showed concordant results, there were also some I've highlighted in the red box here which showed an effect on one platform but not the other. We dug into some of these, and on the right here, we're showing a region of the genome near the GDF15 gene. And this was a signal for a genetic signal with GDF15 protein levels in the blood. And on this SomaLogic platform, there's a really strong peak, a really significant association. But on the Olink platform, there was completely no statistical evidence at all. And actually, that was really insightful. That really helped us. We were using these genetic variants to try and understand if GDF15 is a good therapeutic target for obesity. And if you analyze the results with one protein platform, you got one result, and with another, you got a different result. So actually, we were able to dig into that and to work out what was going on and to work out which platform was likely giving us the right results and therefore what the implications are. So actually, what we found, and Mike Pietza may touch on this more in his protein talk later, is that having multiple measurements from different platforms is actually really useful, because if you only have one platform, you're only getting part of the story and you don't necessarily get the full biological understanding. There were also sometimes differences that we saw between not necessarily the assay platform, but between the people in the studies, or the study designs. So here you're looking at a genome-wide study of genetic signals for glucose levels. On the top plot, you have people who'd been fasted, so they hadn't eaten for usually 4 or 8 hours before the measurement was taken. And on the bottom, you have people who hadn't been fasted before. And you can see there are some quite striking differences in the signals. So some of the genetic regions only seem to have an effect on glucose, immediately after somebody's eaten. So there were some interesting differences that we could find by stacking up lots of studies with different designs at the beginning as well. So I painted a picture at the start of how we wanted to kind of combine genetic data, multi-omic data, and electronic health record data to understand more about the causes of disease. And this is one of our recent works that I don't think we've really presented in this in the multi-omics project much before. This was taking a panel of about 1,000 metabolites that you can measure in the blood, bringing together data on about 20,000 study participants and trying to work out what we could find and what useful insights we could glean. So we found lots and lots of associations between different regions of the genome and these metabolites. And again, reassuringly, there wasn't much difference between we didn't see different results between the studies, which was good. One of the things we learned is actually we only had a relatively small number of cohorts in this study, so we're able to bring together data at the individual level rather than just the study results. And that's enabled us to do some disentangling. And what you see here at the top in this matrix is we have a region of the genome here with different signals for different metabolites. We could actually dive in and try and work out which particular genetic variants have effects on which metabolites. And we can see that some of them, some of the genetic variants, these columns here seem to have signals for multiple metabolites. They have, this one here has 3 different associations, whereas other ones in the same region were only very specific to one particular metabolite. And we needed the individual level data to really tease that apart. So that was a key learning for that. We were also able to identify some causal pathways for diseases. So on the left here, there's a region of the genome that's been associated with breast cancer. We identified that actually we had very similar looking signals. The exact same genetic variants were associated with some of these branched chain amino acids, implicating the PPM1K gene in this region as the likely gene that's causing this breast cancer association, and pointing towards a role for branched chain amino acids in the etiology of breast cancer. So some potentially novel disease insights here that will hopefully lead to better understanding of disease and future treatments. I'm going to skip this one in the interest of time, but another story we thought that was quite interesting that relates not so much to a cause of disease, but potential healthcare need is about chemotherapy. So cancer patients are sometimes treated with a drug called fluorouracil, and it's known that variants in a gene called DPYD, if you carry some of these variants that you see here on the left, you have an increased risk of toxic side effects because the body is unable to break down the uracil in the drug, and in the most severe instances, those can be fatal. What we found by looking at our data is we could find that this variant with this particular ID, RS56, etc., these people do have higher levels of uracil even in the general population, not in cancer patients. And what we found in the statistical analysis on the right was that there was another variant that was much more common in the population, carried by about 12% of people rather than 2% of people. And that seems to explain the effect of the RS56 variant that's currently used in the healthcare system to test for, to try and mitigate these toxic effects. So we're now trying to replicate this in a trial that took cancer patients and treated them with this drug and genotyped them to get their DNA information. So we're now trying to see whether or not we think the panel should be updated and improved to prevent more of these toxic side effects. So other kinds of learnings that we think can come from incorporating the genetic information and the metabolomic information. Those are some scientific successes. I think there's lots of follow-ons from this project, and it's stimulated not only lots of scientific collaboration, but further work as well. So you see here 4 examples of other grants, other projects that have been funded that build on the success of the multi-omics project, including at the bottom the successful renewal of the funding for HDR UK for another 5 years. So various ways in which this work will extend or carry on that I don't really have time to cover much today. Thinking towards the future, what can we do better? A lot of the research that we've done is on cohorts that are white European ancestry British people. So a particular focus for the future is diversity, both internationally, but also within the UK. How can we make sure we're capturing a broader spectrum of people? How can we capitalize on the developments and the evolution that we'll hear about in IT and informatics? And also, how can we perhaps enrich the information in the cohorts? So perhaps with improved health data linkages or other multi-omics information. So I'm going to stop there. And I can see there are some comments in the chat, which I hope are mostly about the PPIE. Sorry if that was a brief whistle stop, Anthony. I can see a comment about having trouble with the graphs. Hopefully I explained most of the acronyms, which are largely gene names, but yeah, anybody got any questions before we move on to the informatics piece? Kathy, thanks.
Yeah, thanks, Adam. What a brilliant set of examples. I'm sure it was a subset of examples of fantastic outputs from this work, but it's obviously been scientifically incredibly productive. So 2 points. One, I'm really interested to hear further, and I'm sure we're going to from Emily, Chris, and others, around the reflections on the kind of future infrastructural requirements, because I think this is really pertinent to the work that HDR UK supports during the next 5 years. You know, how critical is it for individual participant data across cohorts to be pooled in an environment that can support sophisticated led multi-omics analysis, and you know, what are the best mechanisms for us to kind of go about specifying what that looks like, and then, you know, running tenders or whatever it takes to set up something that's there for UK-wide cohorts and their research communities to use. That's one thing that'll probably come up later. The other thing that I'm kind of very cognizant of from conversations related to work with Generation Scotland, sort of wearing my other hat, is how critical it seems to be that cohorts work together to negotiate really great interactions and good deals with industry partners who can provide the kind of multi-omics analysis laboratory capabilities. And I'm seeing some very unpalatable suggestions coming in around kind of exclusive access arrangements that last for years and no one else is allowed to access the data, etc., versus some potentially much more palatable relationships. So I wondered whether there'd been any learnings from that on that perspective during the course of this project.
Thanks, Kathy. Yeah, great comments. Maybe we come back to the IT infrastructure one after Em and Chris, but to say that conversations are ongoing about how we can do that, particularly with Emily's new role. In terms of the omics assays, I think you're right. There is great, potentially much greater bargaining power by bringing together lots of cohorts to try and secure funding or a good price to do assays at the same time. I think one of the challenges we've experienced with that is actually the red tape, trying to, universities don't like trying to procure through one particular university and then sort of subcontract to other universities. So one of the things that we've found challenging is actually how you set up a sort of group purchase in a way that our institutions are happy with, and that the commercial organizations are happy with. Not to say it's not possible, but I guess each additional study and institution brings another set of agreements into it. So that's been one of the challenges, I think, full stop, of doing kind of national cohorts work, but particularly once you start to move samples around or move data around or start to purchase things. So I think that's something we know, for example, from single very large studies, they can get a very competitive price. So there is, I think, more we should do as a community to band together. I think HDR UK is a good vehicle for that. I think we need to work out better how to do it, because I think that's one of the other challenges we face is the agreements and the collaboration, the administrative requirements with a body like HDR UK as well. So I think that's something that we can collectively think about how to improve on, definitely. Good. Anthony, you've got your hand up as well if you have a quick question.
Yeah, I was just going to ask, sort of following on from that. Obviously, data is starting to be much more commercialized now. So how are you sort of pushing back on that? Because that is going forward, that could be a significant issue. And also, obviously, we've got the UK is comprised of 4 different administrations now. So does that make it a little bit more difficult to do UK-wide collaboration?
Yeah, great points. I think on the commercialization, I think there's you'll see throughout these talks there's names of companies who've done these assays, but I think there's also HDR UK has been one of the bodies that's been really vocal in trying to make sure that data is open access. So almost all of the data that you see, for example, from the cohort here in Cambridge, the interval cohort is accessible, and we make it available to people around the world because we want to use it, but we want other people to use it too. And in fact, you often get some really exciting science that comes out that we would never have dreamt up because we allowed somebody else to access the data. I think in terms of the devolved nations, the 4 nations, I think there are some challenges around that in terms of the more in terms of the electronic health record data. And I think one of the things we'll hear about is that maybe kind of putting all the cohorts together in one place is quite a challenge, but we can do better than just each individual cohort. So maybe on that note, I'll hand over to Emily Jefferson, who's one of the co-leads of the informatics infrastructure work package based in Dundee, but now has a role as the Chief Technology Officer for HDR UK, so is now kind of actively working from the HDR UK side on solutions to these kind of challenges as well. So over to you, Em.
Thanks very much, Adam. I'm just going to share my slides. Can everybody see that okay?
It's coming. I'm just got a black screen at the moment, but it says you've started screen sharing. Okay.
Oh,
it's going to complete.
Sounds ominous.
It's good to introduce a bit of tension to these things.
Oh, it says it's still loading. Ah, that's unusual.
There we go.
Can you see now?
Yeah, we've got it.
Fantastic. Okay, great. So what I'm going to do today is give you a little bit of a whistle-stop tour of what we did on this multi-omics consortium. Okay, fab. This is the progress through the talk today, and I'm just going to talk through each one of these different things, essentially talking about a journey to where we are today. So as Adam said, we were looking at the informatics infrastructure, looking at scoping, discoverability, and an analytics platform. So the way I view this is a bridge across 2 different worlds. You have the world of bioinformatics, where you have large-scale genomic and imaging data and things like that. They're openly, freely shared. And on the other side, you've got electronic healthcare records, which have to be used within trusted research environments, and they're usually kind of quite small and previously locked down. So what we're trying to do here is bring these 2 worlds together so that you can have one environment where you can do all of these different things in one place, helping streamline research. So the first thing we did was try to understand the requirements of people. We know about how to run TREs. They're usually there for kind of epidemiology research using standard stats packages. So for those of you who kind of don't know about that, just imagine like a complex version of Excel. But when you're looking at these other sorts of data sets, you've got things like data volume, massive data sets. You need to be able to program within those environments. You need to be able to install a range of different tools that aren't stats packages. You need to consider the cost and the cost of the software as well as the hardware. You need to have different types of hardware than you would normally have in a traditional TRE. So you need to think about things like high performance computing clusters where you've got lots and lots of CPUs, and you also need to think about things like GPUs as well. You need to think about how you might want to export an algorithm if, let's say, you've been working in the field of AI or predictive models. So I kind of class these as next generation TREs. They're taking us to another level of what is capable within this space. So we sent out a series of questions to the Multi-Omics Consortium to ask them about what they wanted to see within these environments. I'm not going to go through all of these, but just to let you know the sorts of questions that we're asking. And this informed our design decisions. So then we said, well, somebody else in the world must have done this. Let's go and see what else others have learned and see if we can engage with them and collaborate and create a better solution. So we interviewed 22 different national and international TREs. And what we found was that they generally adhere to something called the 5 safes model, which is all about how a TRE can be locked down to make sure that the data can't leave. Most of them, however, in fact, pretty much all of them, did not support next generation capabilities. So things like different data types, like the genomic data, ability to support artificial intelligence, ability to install your own software within that environment. They didn't support handling of big data, and they didn't import automation around timely import and export of data outside of those environments. And most were not able to do things like supporting federation or interoperability across them. So, conclusion, everyone is struggling with it. So when we were struggling with it, so was everybody else. We were actually kind of forging forward in this new land to faster pace than others out there. So there wasn't really anything that we could really learn from out there. So then we had to decide what did we want to do. So we had a legacy on-premise TRE environment, trusted research environment, that would not meet the requirements. So we did a cost-benefit analysis. Sorry, I've got a cold, so I have to suck my cough sweet every now and again, otherwise I'll have a coughing fit. So we did a coughing benefit analysis of the public cloud versus buying an on-premise solution. And the amount of time and effort actually that we required to put into that should not be underestimated. It was a huge amount of effort to really properly analyze these things. And what we found was that our estimation of a cloud solution was that it wasn't going to be too much more expensive, but it would give us a range of additional capabilities. So it would enable us to scale on demand, so just ask for more compute power as and when we needed it, rather than having to buy a machine, install it, and configure it. It would enable us to use things like high performance computing and GPUs. So that's huge numbers of CPUs and GPUs set up so that you can analyze this sort of data. It enabled us to use software that had been pre-configured, ready to support a range of different types of modalities of data. And so there's a load of software within cloud environments, and you can just basically tick the different box, and that software can be installed within your environment. And it also reduced the staff time maintaining the system. And that was really quite important. So we looked across a range of solution providers, and at the time, the Amazon Web Services solution, which is one of the public cloud offerings, had features which made it more attractive at the time than others. And so we formed a partnership with AWS to co-create our trusted research environment solution. So, designing the solution, excuse me. So our on-premise environment, or on-premise environments in general, include multiple compute nodes connected to a high-speed network, and multiple people share these compute nodes. And this is a bit of a challenge because different jobs from different users can run on the same compute node, and temporary files or cache data might be left behind and could be visible to others. And so this would not be suitable for a TRE environment. And so now what we're doing is putting this high performance computing environment in the cloud with unlimited compute, with no sort of queuing. You can just ask it straight away, and you don't have to sort of share that with other people. You've got dedicated clusters per users, full encryption, and automatically stop and use compute so that you don't waste money on these sorts of things. So I'm sorry. We were really delighted that we got some additional funding for this, a capital award from World Class Labs through HDR. And it meant that we could really scale the work that we were doing and our partnership with AWS. And so we created the TRE environment, and we configured it with something called Amazon Web Services batch processing. And this is a service which basically allows you within your T environment to send off lots and lots of jobs to a range of different computers out there, hundreds of thousands of them if you so desire, all at once in service, so that it can run these sorts of massive computational tasks. And so we worked with AWS to configure that. And we also set up a different capability in there using what's called Nextflow. So Nextflow is a pipeline which allows you to reuse research packets of software that enable you to use a range of different tools and a range of different suites of software, and run these suites of software on your huge number of computing machines all at once, and coordinate that activity. And it enables you to reuse software that people have already built in a really easy packaged way. And so we then applied this new infrastructure to 3 different exemplar projects. So the Go Fusion cohort is a really large cohort in Scotland, linking lots of separate cohorts together, including the share cohort, and it's got SNP data combined with patient data, and it's also got legacy data from a cohort called Go Darts, which is both diabetics, and then we've added non-diabetics, and we made this whole set of data available for an MSc course for training and developing, so that people could actually use this environment live in analysing all of that data. We also enabled something called GoDB, which is a database with a lovely front end for searching across multiple chip platforms. So as Adam said, sometimes the results come back quite differently depending on the assay that you've used. And this actually sort of pulls out where there are differences across different assays and helps you analyze them and drill down into that. And so we installed that within the TRE environment, and it also linked then to the underpinning VCF data. And another project that we looked at was not just human genetic data, but we also looked at bacterial genomic data, and that was then linked to human electronic health care records, so that we could compare people's healthcare records with a particular type of bacterial infection that they had. Oh, sorry. So for the purposes of time, I'm just going to quickly run through this. We had an on-premise environment that we basically enhanced with lots of additional capabilities in order to handle these sorts of data sets. The working examples were set up, and we found that it was a much better, faster solution. So we did a feasibility study, and we asked people what they thought of the environment and how easy it was to use. 1 out of 5, basically most of them ranked between 1 or 2 where 1 is easy. So we've now taken it to another level, and we had some additional funding for a DARE sprint project, and this is called Treehouse, where what we've done is taken the design of the architecture that we've built, and we've now made it freely available for others to use, because we don't want this to be a closed system that nobody else can take advantage of. We want other people to be able to build such an environment within the cloud and reuse that. And so we're sharing this code with others for people to adopt and use. We've then moved on even further, and we've looked at another DARE Sprint project, a later one, where we're looking across a range of different TREs to create standards across different cloud solutions. And that's across the whole of the country, bringing together several different ones to create a reference architecture. And that works not only across health data, but also many other different types of data as well. And so we've got an ongoing project for the next 9 months, where we're working across all of these to essentially standardize this so that more people can utilize such infrastructure going forward. And we're going to make those sorts of things explainable and share the outputs from that environment. And I'll just very, very quickly finish with another DARE project that we did called Grey Matter. This is all about artificial intelligence being developed within such environments. And essentially, if you're wanting to take out trained algorithms from a TRE, it's really difficult to make sure that they don't contain any potentially identifiable data. And so TREs don't really know how to do this. They don't have mature processes to do it, and they're not really engaging with AI because they're worried about it. And there's a range of different risks associated with that, but I realize we're running short of time, so I'm not going to go into those. If you're interested, please do chat with me, because there's some interesting challenges there. So we released something called the recommendations for TREs, a green paper suggesting to TREs how they might want to put in the associated controls so that we can actually handle that sort of processing and make these next generation TREs. These are all of the brilliant people who worked on this project. Just so you know, HDR, we're recruiting for some new roles. I'm always going to use an opportunity to promote them. So please do let anybody know that you're aware of that might like to come and work in the new team. And thank you very much for listening.
Brilliant. So many achievements there across different projects, which is great to see. And I still really like, I think, you know, what you've really done in that project is bridge those worlds, is trying to understand, you know, I think that key piece about understanding what's needed. So many of these solutions are out there that we could have tried, but actually working out what the pros and cons, what's needed by the community, and that kind of ongoing dialogue, as we talked about in response to Kathy's question earlier, of continuing to try and work out what's best for the range of different projects you're supporting is really crucial. I suggest we hold questions until after Chris has talked as well, because I think there might be some commonality of question based on the approaches and things they've tried as well. So Chris Orton's from Swansea, and co-led the work package with them, and has also tried various solutions and tried to understand the needs here. So Chris, the floor is yours.
Yeah, thanks very much, Adam. Hopefully everyone can see that okay.
Yeah, that looks good. Thanks.
Brilliant. Yep. So morning, everybody. So I'm Chris Orton. I'm a program manager based at Swansea University. My main role over the last sort of 10 years or so has been developing research infrastructure that uses Swansea University's architecture, which is called the Secure eResearch platform. I'm also the upcoming technical lead of the Inflammation and Immunity Driver program within Health Data Research UK's second 5-year period, the Quinquennium 2. And essentially, we're hoping that a lot of the work that Emma's already very eloquently described, and hopefully I can do just as good a job, and all the work that I'm going to describe will be reused and amalgamated moving forward, so that we can help the community, as I'm going to describe, in moving from molecule to health record analysis and linking the pipeline together. So I think this is being adequately described, so I won't go into too much detail that everybody else has had, but essentially the challenges that we're all facing is that there is a massively complex pathway from the sample and tissue collection that happens in labs, particularly with the cohorts that are part of the consortium, but generally with cohort studies across the UK, through to deriving omic data from that. Most traditional TREs, such as that as we run in Swansea University, such as the SAIL Data Bank and the rest of the Secure eResearch platform tenancies, specialise in housing structured data, such as health records, observational, clinical data survey, for example. And that isn't, as Em mentioned, particularly useful when coming to look at large unstructured molecular data sets. This has also been combined with the fact that specialist TREs and infrastructure such as that at the Sanger Institute, EMBL, EBI, etc., have traditionally been the leaders in dealing with complex semi-structured or unstructured data because of the dedicated hardware and software needs that needs. However, for sharing that data, changes and obviously advances has had to be done, and we're trying to, between myself and Emily and obviously the rest of the community, bridge the gap between these two ends of the scale. And obviously make sure that they are complementary in the way that they work. One thing that we've noticed as well is that generally on the health record end of the scale, infrastructure prerequisites coming out of the NHS or out of the data providers have been particularly obstructive for open sharing and for sharing across platforms, generally because they've been difficult to standardize around environments. So for example, standards, accreditations, etc., not every single researcher is able to attain these, whereas of course large scale organizations are now starting to put more of their time and effort into actually getting these accreditations to make sure we all have the same standardized gold standard for how a trusted research environment or general research environment, secure data environment should actually run. The other thing that we've got to bear in mind coming at this from the opposite end of the scale, so rather than the research that Adam showed earlier, but the platform provider end of the scale, is we have to bear in mind that the skill sets are very different between statistical analysts and those who are doing the molecular end of the the work in research projects, and therefore the flexibility is really needed in the operating system, and particularly having an absolute gamut of software available for these teams to be able to work together. One thing that we also bear in mind is that not only providing the platform is part of our mission, but also enabling pipelines to be built to structure data that can obviously make the data more usable for a wider audience, but also let the specialists do their thing as and when appropriate. So just to give a few examples of 3 tenancies of the Security Research Platform, which I'll go into slightly more, which are trying to achieve this, of which include the cohorts of the Multi-Omics Consortium. So in Swansea University, we have a department called Population Data Science, which is part of the medical school. And essentially anything that is to do with population health, we are interested in. And so we have partnered with a number of initiatives which are looking at large-scale population cohort studies who make use of a tenancy of our infrastructure to create their own governance, their own operating procedures, enabled to develop data. So what we've done in the same way that Emma's spoken about earlier is essentially interview our data providers and their researchers and see exactly what they needed to actually make their data sharing pathways easier, and obviously in this space develop omic capability for analyses. So just giving these examples here, Dementias Platform UK is probably our most mature in the cohort space that we have in Swansea University, and this is led scientifically by the University of Oxford, and Swansea University leads on the technical side of things. So this is a collection of over 40 longitudinal cohort studies from across the UK and some international providers as well, who have deeply phenotyped their participants, some over a vast amount of years. Some are from birth cohorts going back even to the 1930s, when the participants were born. And essentially their data collection has been periodic since then. And obviously as things have developed, so has the technology that has collected their data. And essentially some of those have been able to take samples, and quite a large majority of them have been able to take samples or clinical subsets from their participants, and that has been able to derive genomic, metabolomic, proteomic, and obviously massive phenotype data on these participants. Imaging is a massive area as well that we're also driving forward, much as the Pictures project that Em spoke about up in Scotland. Given that this is a neuroscience angle, brain imaging is a large part of what Dementias Platform is trying to do. And 4 of the HDR UK Multi-Omics Consortium cohorts are part of Dementias Platform UK. The second is UK Longitudinal Linkage Collaboration. So this has actually only been live in the last couple of years. This has come out of COVID-19 and essentially was an all-hands movement to combine 20 longitudinal cohort studies, creating a master cohort spine to link them to routine health records, NHS Digital, Sale Databank, Scotland, Northern Ireland, where relevant, and essentially create a super cohort that was able to perform research on at scale, particularly needed for COVID-19 because of the granularity of the data that each of their surveys and health records can provide. And what we're trying to develop is, in association with all the other work that we've done through this project with Dementias Platform UK and with Sale Databank, is to provide an omics work package to them to replicate analyses that can happen in these other tenancies, and of course, hopefully further develop from what can happen in their own jurisdictions. The final one, which comes out from the health record side of things, is the Sale Databank. So this is the trusted research environment hosting Welsh health records. And what we have tried to do is we're trying to move the other way, trying to incorporate genomics into our health record hosting, which is 20 billion plus rows of data, and make this available from a more routine health record side of things. So this is lab data coming out of the NHS, genotyping and imaging coming out of the NHS, but obviously very linkable to the participants of any study that may be based in Wales, or if we were hosting a cohort study from the UK, simply on that infrastructure. So just very basically, this is a top-down view of what SERP is. So Security Research Platform based at Swansea University is a publicly funded research cloud. And as my Chief Technical Officer would say, the cloud in our sense here is not something like Amazon Web Services or Azure, for example. This is a fully on-premise system. And at the end of the day, cloud is simply somebody else's computer where other things happen. So this is operated on a cost recovery basis. So all of the work that we do is operated to simply pay for the part of the system that is being used, and obviously recover costs in order that we can recoup that and buy new infrastructure as that is happening. And as I've been describing, this really is evolving based on need. The whole premise of SERP is that this is multi-tenancy. So for example, Sale Databank is the flagship. It's been there 15 years. It's been there the longest, but now we have over 20 other tenancies who have been able to reuse the model of the infrastructure to bring data in, share that, apply their own governance model, and get that out to researchers. This also protects the provenance of the data and making sure that data providers always remain in control of the data they share. Now, for example, this infrastructure powers Sale Data Bank, Dementia's platform, UK LLC as mentioned, and the HDR UK Northern Ireland site. So it's a very busy slide, but this just shows you a few examples of who is currently using our infrastructure at Swansea University. And then if I just move this on very quickly to this one, the ones that I've highlighted are essentially ones who would be running into issues in the space of molecule to health record analysis. So it's a vastly applicable issue that we're all trying to address for an absolute, vast array of different research organizations. So it's not insignificant at all. So very briefly, and again, this is actually a simplistic diagram of what UK CERT looks like to an end user, and there is a version which has got cables and networking all over the place, but I'm not going to go into that today. But essentially, this just shows you how our trusted research environment works on the flexibility side of things, in terms of not only are there flexibility at the software end, which is on the far right of the screen, in terms of what type of database you're using, what type of application you're using, which are the yellow boxes on the far bottom right corner, but also in the way that you actually connect to the environment. For example, there's a way we can make this available to people using incredibly sensitive data, which could be just inside a building. They're only able to access it there. Our most common clearly is you access that through the internet, which is the globe at the top on the left. And then obviously we also have the ability to interface with the NHS and allow clinicians to actually log in and use data as well. So just showing a very brief path of how somebody might connect through the internet into DPUK, and the permissions essentially on our system allow somebody to go into the relevant part they're actually able to see. For example, you'd never see all three of Sale UK LLC and DPUK with one account. You'd only see what you're allowed to see. And the thing on the right hand side, I've circled object storage and data lake, because in this space with omic data, that's most likely where it's going to sit. And essentially we want the flexibility of people being able to go between Linux VMs, which are traditionally most used in the omics world, through to Windows, and obviously the statistical end of the bargain, which is what I spoke about earlier. So using the funding, particularly from the Multi-Omics Consortium project, as I mentioned earlier, one of the most mature TREs we have is Dementia's Platform UK, but the Security Research Platform environment has been able to provide upscaled compute, so that is at the top in an imaging sense, so not directly through the work that the cohorts have been working us on, but this is an exemplar project with the Alzheimer's disease data initiative, which has also been part of an HDR UK initiative for COVID-19 called iCoda. And the main two we've been able to move forward essentially are the HPC cluster capability, which, as Em mentioned before, is a really difficult thing to tie down when you've got hundreds of people hammering away at it, when you need to compartmentalize this. So essentially making the compute available to people, having the hardware available is simply not enough. We've also had to network in there job scheduling to make sure that there is a logical manner in which people are hitting the stack, but also stuff, as Em mentioned earlier, Nextflow, Kubernetes, Docker, etc., so we can actually containerize people's compute and actually parallelize this as well to ensure that multiple people can work using the clusters at the same time. One of the largest things that we've been able to, or at least trying to move forward, is in the linkage infrastructure as well. Now, this isn't just between health records and cohort data, but this is between health records, cohort data, and the omics generated, and ensuring that one person's ID or one person is actually traceable. And obviously, at population scale, you can then link everybody through from molecule to health record. As you can see on the bottom right, Airwave, Epic, Norfolk, Generation Scotland, a UK biobank feed into Dementia's Platform UK and are sort of feeding into every single part of this pipeline on the multimodal data analysis we're supplying. And really the main message here is none of this works without having the appropriate staff to help with this. So essentially the compute does a certain job, the environment does a certain job, but you also need to bear in mind that data management is a huge task here in getting the data ready for use by different levels of the community, but also in ensuring that it all works and that the back end feeds the front end and that the results coming out therefore equal good science. So just moving forward in partnership with MRC Climb, which is the cloud infrastructure for microbial bioinformatics, we're looking to further this pipeline of essentially containerized, parallelized analysis, which means that multiple researchers can run their research at once across any of the tenancies we're working with. And the last thing I'll mention moving forward, going into this, is we're really hoping that all of this will feed into the federation developments that we have coming up, particularly through the DARE UK theme and into the HDR UK QQ2 work. What we would like to see, obviously, is that given that this is a massively complicated part of it, we start with the health record end of things, branch that into the omic data by enabling complex analytical packages to move to the data, and in some cases allow data to be visible through a single interface for this work to actually take place. So we believe we've actually been able to move this forward despite the pandemic quite significantly. There are lots of people trying to achieve all of this, and really it won't be a success unless we all speak to each other and keep this moving in a coordinated direction.
Brilliant. Thanks, Chris. I think that last take-home message is really key. It's been amazing to see how much the field has moved on in the last 2.5 years during the course of the project. And of course, in one way, that's a challenge to sort of keep up with what's the latest optimal solution. But on the other hand, there are so many opportunities now. And I think that federation opportunity you talked about, there's 4 cohorts that are already in this environment and analyzable. And then there's another 3 over in this environment. And we've already alluded to the perhaps the different health record linkages in different places as well. So I think this idea of federated access is going to be increasingly important, and is one that HDR UK is very actively working on, which is great. Any questions then for Em and Chris, or things about the IT and informatic infrastructure for cohort solutions? Kathy, you've got your hand up.
Yeah, a sort of simple question for both Emily and Chris, because I think they both touched on this. So we've talked about how most TREs deal with and handle quite well structured data. So that for the lay members, you probably have already got your heads around this actually, because you've been learning on the way, but structured data is data that could be represented in kind of coded form in a spreadsheet as a bunch of ones and zeros, or specific codes aligned to items. So it's much, much easier to cope with. And we've been talking here about various types of unstructured data, different types of omics analyses done on samples, and imaging data are probably the most prominent. For both of those data types, essentially, and this would apply to free text as well, we need to surface structure from them to be able to then integrate them with the already structured data to perform analyses. So how much can that surfacing of structure be done separately in different environments using a common set of instructions, and then one kind of links the data for the ultimate analysis? Or how much do you need the information from the existing structured data to be able to do that derivation?
Do you want to go for that one, Chris, then I'll follow?
Yeah, sure. Yeah, it is a massive, as I think I mentioned, it's not an insignificant data management task, and I think it's actually part of something which doesn't actually get funded very often on research projects, is bringing out, as for example, with the Dementias Platform UK project, there's a lot of work in image-derived phenotypes, essentially not just analysing the raw images that are being provided or even a format that has been standardised, but actually analysing it to then bring out information that can be then used in combination with other types of data. But it's not something that is routinely done, I think, across the board in these spaces, but it is something that would definitely accelerate the way that the data can be combined, particularly because the formats of the data could be made easier to use, particularly as things like federation do come into being, where standardizing file format structures, that sort of thing, will be of massive use to enable you to scale up across all the environments. And that's exactly the same in genotyping as well. We have cohorts who obviously do the work for us, which is marvelous, because they're able to actually derive the genotypes themselves and provide something in what would be maybe flat file format, and then that can be combined with all of the other survey, observational, clinical data. But it's not something that's always done, and it's not something cohorts always have the capacity to do. So it would be good to see if at some point there was maybe an army of people who were really interested in doing that piece of work. And I think it's definitely something to bear in mind for capacity building.
Yeah, so just following on there, so echoing Chris's thoughts. I also think that there's a lot of standardization of the of the biological data due to the fact that the community have been working together freely, sharing tools, sharing methodology, sharing summaries, sharing formats in a much easier way than we've been able to do within the world of sensitive data. And so I think there's already a lot out there that we can build on. I think it's a case of surfacing those things that already exist, rather than trying to reinvent the wheel here. But I think there's a lot more that we can do. So Adam and I chatted, I don't know, it's about a month or so ago now, about the idea of actually trying to capture some of this multi-omic data as metadata within the gateway, so that you can actually search for that sort of information across the UK as well. And that would be a really helpful thing, so people can find the cohort information out there as well.
Great, thanks. Andrew, I can see you've got your hand up as well. Welcome, and feel free to ask your question.
Yes, thanks, Adam. And apologies, please excuse my lateness, but I was at the Public Advisory Board, and so I'm sort of juggling stuff. And this may have been picked up. So my question is around connectedness and our quest to have a single map that we all point to. Because what I see here is convergence of initiatives across the UK from a sort of grassroots, right? At the same time, I see stuff happening with Genomics England, UK Biobank, Our Future Health, and we've got folk like DNA Nexus, LifeBit. So how close are we to having a single map? And are we sufficiently connected with specifically Biobank and Genomics England, because I think we're going to see a major play on that. And then the second question, it was really stimulated by Hans Erik's presentation on Tuesday. Authorization and authentication. Is there an opportunity for us to land on the GA4GH standard just to demonstrate its utility? And I say that specifically. My third question, if you'll permit me, is currently GA4GH are looking for driver projects, which are international. Is this an opportunity for this team to think through what would a GA4GH driver project look like? And it might gear us up internationally. So, sorry, 3 questions, and I was late, so excuse me.
Thanks. Yeah, we've actually already answered all of those questions previously, Andrew.
Nice try. Nice try. When you say map, Andrew, you mean, I guess this would be some kind of where can you access each of these different data sets, the UK Biobank, the Genomics England, given that they're in different environments, and how do you interface between the different environments?
So a map, or even a forum where we're discussing this stuff, because I do see them as being major players. So.
Absolutely, yeah, and I think that's part of the challenge. Is we know that there are some major players in the area that have developed their own systems, have their own solutions, and they won't necessarily interface directly with the rest of the cohorts across the UK. So I think this idea of the federation, as the analyst, I want to be able to. And some of the sprint projects have exemplified this. I want to be able to run the same piece of code, ideally, across different cohorts, even though that data is held in different places around the country. And I think that federation idea, that connectedness, is going to be crucial for that. But Chris, do you want to chip in on the other questions, which are much more your domain than mine?
Yeah, so just in terms of the federation component as well, I think there's some really exciting things that we can do linking this driver to the federated analytics workstream for the next 5-year strategy. I think we've already sort of started in those sorts of conversations about how we could do that. I think, again, the gateway, if we configure it in such a way, can actually be enhanced to provide a map that's a technical map around all of these different things. But absolutely, we need the forum to bring everybody together. In terms of the GA4GH driver, I think that would be a really exciting idea. Very happy to look into that. And in terms of the identity components, actually, I was having a conversation with Simon Thompson at the technology ecosystem meeting the other day, talking about which standards and things we wanted to investigate, and whether what stage of maturity these things are in order to look at adopting them. So there's already talk about a program of work to look into those things.
Great. John, you've had your hand up and waited patiently for a while. Do you want to come in with your question, please?
Great. Thank you. And thanks for the energetic and illuminating talks throughout the morning. So question really for Chris, possibly Emily. So a refrain that we hear from leaders in the genomics molecular world, Matt Hurls, Hilary Martin and others, is that many of the infrastructures that we have now, possibly SERP, designed for health records first, and perhaps as molecules, genomic data as secondarily, and trying to retrofit, are still too clunky to be fit for purpose. To what extent do you think trying to retrofit things that were designed for health records first is the way to go versus using bespoke solutions, things that are designed for the molecule to health record seamless journey?
Yeah, and I think we'd be the first people to agree to a certain extent that the health record infrastructure is not something that's suitable for running molecular analyses. But I think rather than retrofitting the exact architecture that we have set up for health records, it is about actually bringing those bespoke solutions in alongside them and networking them to an extent where they can actually talk to each other. So those that need to do the large scale health record analyses can still do that with the architecture which works well for it. But at the same time, you can then burst in whatever way you need to the levels of compute and to the levels of software and the different combinations to be actually able to do the genomics or other omics analyses that you can. Hence why I think if you look at Emily's presentation and mine, there's a lot of the same products that are being used in this space to do this work, whether it's hosted on AWS or on-prem or whatever you want. It's very similar architecture. Hence why I think, actually moving to Andrew's point earlier as well, we are actually all starting to coalesce around a single roadmap about how this was worked. And I think as part of the technology ecosystem, our discussions really are whether we need to continue being the ones who retrofit ourselves, or whether through federation, data that needs to be hosted by, for example, ourselves, who may be hosting, more mature health record data that then needs to link to the omics, could federate that work to groups like Genomics England or others who obviously have the expertise in the space of dealing with the raw data at source. And I think there are projects now which will allow us to actually do that, either through the DARE initiatives or through the fact that we're all coming around HDR QQ2. So I think those are sort of pathway decisions we all have to make. But I think what we're trying to do is obviously help the customers and the people that we work with at the moment as best we can to actually branch out and do this all in one space. And if we can make it UK wide, then all the better.
Thanks, Chris. Thanks. Lots of questions stimulated by this important topic, and the way that we go forward analysing these data. There are some more appearing in the chat as well. In the interest of time, I'm going to suggest that we draw this session to a close and give everybody the break that I promised at the start, which we're now somewhat late for. So please could I ask everybody to reconvene at 11:25. So in 15 minutes time, stretch the legs, get some caffeine, whatever's required. Maybe Em and Chris, if you could tackle any of the outstanding questions in the chat, or even others may have insights onto the genomic data breaches question in particular. So thanks for a great session. Thanks to all the speakers and for everyone who's commented and asked questions as well. Keep the questions coming, and I will see you in 15 minutes, please. Welcome back, everyone. It's 11:25, so we'll start again with the second half of the morning's program. We're going to hear, as I said at the start, more about some of the science that's gone on in the metabolomics work package, the proteomics work package, and then the work package that looked at trying to integrate across domains. So I'm pleased to welcome Professor Joanna Tsoulaki from Imperial College, who's going to talk to us first about particularly work on the metabolomics side of things. So Joanna, over to you.
Thank you, Adam, and thanks very much for organizing the day today. It's been very interesting to hear all the work. So I'm going to talk a little bit about metabolomics. And Adam talked a little bit about that too, as we shared the work package on it. So if I do like a brief introduction on metabolomics, the reason why we spend a lot of time and energy and work around the integration of metabolomics across cohort is that it is one of the most challenging technologies to integrate across different studies. What they are, metabolomics, they are the assessment of measurement of low molecular weight metabolites in different biological fluids. And they are interesting because they provide a comprehensive assessment of gene actions, intrinsic metabolism, and environmental exposures. And by that, we can look into molecular pathways that underlie complex biological processes. Now, we can measure metabolomics in blood, but some studies measure in urine or in other biological fluids. So that creates one level of difficulty of integration across studies. But apart from that, we have different platforms, different technologies to measure them. You see here an NMR machine and a mass spec. So these are the two main technologies that we can measure metabolomics, and they measure different metabolites. We also have different modes, so we can have targeted modes where we measure a pre-specified number of metabolites of known chemical identity, but we can also run these technologies untargeted agnostically to measure different features, which we then identify. And targeted metabolomics may be more interesting because we may identify an association with a metabolite that we haven't thought before to measure, but they have different challenges. Sometimes we cannot identify the metabolite of interest, and it's much more difficult to integrate them across different studies. And of course, we have problems around statistical power with that, the generalizability of findings, reproducibility, etc. We have two main technologies, the main companies, Metabolon, which measures mass spectrometry, and they provide a comprehensive panel of a number of metabolites in MS, and many large cohort studies that were part of our consortium had this measurement. There is also Nightingale, which has a well-known NMR panel of metabolites. Again, a lot of studies have this kind of data, including UK Biobank, but there are also other companies like Socrates or the National Phenome Center, where we have an untargeted panel or sometimes targeted panel that come from different protocols. There are also initiatives like the Consortium for Metabolomic Studies to bring those together and to bring epidemiological studies together with metabolomics data. These are mainly focused on bringing together data with the same studies with the same kind of data, so either Metabolon or the NMR. I have an example here of a work we have done on trying to harmonize and integrate untargeted MS data across cohort studies. This is a paper that was published last year, which is more an analytical, it was published in analytical chemistry, and it is an analytical chemistry type of paper. MS perform high resolution analysis of small molecules. And we had thousands of participants here, but in different cohorts. It remains challenging in large-scale MS to bring together the data and combine or compare data sets that are generated from different analytical methods, instruments, or softwares. What we have in common is the retention times, the mass to Z ratios, and also the feature intensity. And we used this information. So from each study, we had this information. Say, for example, we have here two different studies, and we try to match the features from one to the other so that we can bring those features together and analyze this matched now mass spectrometry data set. So we provided here in this paper, which was a work run led by Rui Pinto, a three-steps procedure in order to match the two different MS data sets that were coming from the MESA study and the Rotterdam study, and later on with the Airwave study. And first, we had an algorithm where we could find all possible matches. Then we wanted to find unique correspondence between those matches. And then at the end, detect and exclude those that didn't match well. And we validated this procedure. We applied this to a data set which we haven't used to come up with the algorithm, and then we've sort of at the end annotated and found the chemical identity of the match metabolites in each of the 3 cohorts, and we were able to validate that the matches that we were doing were correct. And we presented also a tool in GitHub for others to use that for the integration of large-scale MS data across studies. Now, moving on, what else we could do? Now here there is a graph which is meant to show you, for example, in Airwave, we had different subsamples of the study having metabolome data, or having the National Phenome Center NMR data, or having the National Phenome Center MS data, or having Nightingale data. But we also had overlap between them. And what we are trying to do is use the overlap between the samples and use the information that we have in this overlap to predict the values in the rest of the samples, treating them as sort of missing values. And this is work led by Alex Smith, who is fellow in data science, trying to use the information of data in this sort of overlapping circle in order to predict the values of, for example, metabolome data in this set. These are some results. I mean, I don't present the actual method that Alex used, but some sort of high-level summary. You can see here the UMAP plot, where indeed the metabolites, the features clustered together from the same pathways, giving us some evidence that this method works well. Or later on, we have this imputation performance of metabolome features that are grouped by categories. And what we saw is that we were able to predict lipids, for example, much better than the rest of the features, the group of features, probably because the National Phenome Center assays were lipid-based, and this is where we had better information. So the model was able to sort of perform better in predicting that particular category. And then, as a form of validity, we tested associations between the imputed data and the real data, for example, with body mass index or with C-reactive protein, and comparing the blue and the orange dots, we could see that our model, the imputed data in a subsample that didn't have this data before, works as closely with the real data, the rest of the data that had true measurements. And we can use this type of approaches sort of to maximize the potential and to maximize the data sets that we have, as well as to harmonize data within cohorts and between cohorts. So we can extend that algorithm between cohorts as well. Now, what can we do now that we sort of managed to integrate the data sets and match the data sets? And here is some work that Abbas Tenhan led from our group, having data from 3 different cohort studies, including Airwave, Rotterdam data, and the Finger trial. And using the algorithm that I presented at the beginning, we were able to harmonize the metabolomics data. And then our interest here was to look at associations between genetic variants that we knew are associated with Alzheimer's disease from large genome-wide association studies, but we didn't necessarily know why they're associated with Alzheimer's disease. In other words, what's the mechanism through which they may increase the risk of disease. So for each one of those SNPs, I think there were around 90, we ran a metabolome-wide association study. So we associated each SNP with the range of metabolic features that we had measured. And here is, for example, a Manhattan plot showing each section here is a different gene of the SNP, and these are association with metabolites. You can see that we found a lot of association with a particular SNP in the APCA7 gene. And here with the red line is the false discovery rate. So our statistical threshold, we have quite a few associations, all with metabolites that are close to the lactoceramide pathway, so a particular lipid pathway of interest. And if I go back to this graph, we took that analysis further. We looked at associations. First of all, we annotated the metabolite to lactoceramide, and then we sort of do Mendelian randomization to check the causality, the potential for causality of this association, other associations with other phenotypes, and then some experimental work to further add validity of the pathway. And this is here the suggested pathway. So we were able to go closer to a potential pathway that is important in Alzheimer's disease. Here, for example, are different SNPs, or different genetic variants within the APCA7 locus, and their association with metabolites. And the circles represent different metabolites that are measured by different platforms. So, for example, this is metabolon, and this is the HeLiC positive, lipid negative, and lipid positive assays of the National Phenome Center data. And the associations that come with all those SNPs within the same locus on lactoceramide-related metabolites and pathways. And these are the results of Mendelian randomization, which support a causal association between this particular metabolite and Alzheimer's disease phenotypes. So moving on, we can sort of use these approaches as well to study genetic variants, metabolites, and diseases, phenotypes of interest. Here is some ongoing analysis specifically on the ApoE locus, that we know it is an important locus both for cardiovascular disease and for Alzheimer's disease. But we don't know necessarily that the pathway between the gene and the phenotype between Alzheimer's disease and cardiovascular disease is the same. So it's unclear if the gene affects multiple diseases through the same or different pathways. And we have here the SNPs that are important for atherosclerosis, for coronary artery disease, and for Alzheimer's disease within ApoE. The disease, and between them, the metabolic pathways, the metabolites that are associated with them. And we can see that there is there are some shared metabolites, but overall there seems to be disparate disease pathways, not necessarily shared between the SNP, the genetic variant, and the disease. Finally, we can look at the categories of metabolites. This is a paper we had last year in PLOS Medicine on fatty acids. Fatty acids have been of interest to us many years in epidemiology as potential supplements for reduction of the risk of certain diseases. Having large-scale metabolomics data across cohorts has enabled us to study well the genetic variants that are associated with fatty acids. And indeed, if we group them, if we look at the different categories of fatty acid, we can have their own genetic variants. Some of them are shared, some of them are unique to different fatty acid components. And having this genetically predicted fatty acid level, we can look at associations across the phenome. This is sort of a phenome-wide association study across all those 8 categories of fatty acids. And then we can look deeper in the results, do Mendelian randomization, and see which phenotypes seem to have potential for a causal association, with different fatty acids. We did find associations, protective effects of particular fatty acids with obesity and cholelithiasis. But perhaps most importantly, we did not find supportive evidence for a decreased risk of heart disease. So the data did not support the supplementation of unsaturated fatty acids for cardiovascular risk reduction. And I think that was it, flavor of the work we have been doing on this area. There is much more to do, but I think the first wave of the results are really promising. Thank you.
Thanks very much, Joanna. Brilliant. I think you really exemplified there some of the solutions to some of the challenges, particularly we touched just before on the idea that some of the omics data, molecular data is often very structured, but actually all that untargeted metabolomics data from different platforms, trying to see if it melds together is a real challenge. And I think you've really nicely shown in the later examples there how using some of the metabolomics data is filling in the gaps between DNA and disease and helping us understand causes and causal pathways. So really nice. Does anyone have questions for Joanna? Feel free to pop them in the chat or raise your hand. Maybe I can just start with a question then. You kind of showed in terms of the integration across the different platforms initially, the UMAP plot to show that largely the similar species were clustering together. Have you done the same kind of analysis that you showed, say, with the BMI and CRP, where you're you can then test to see whether there's similarity of association across the different platforms? So they seem to cluster together, but do they give you the same answer or not?
We are, this is ongoing work, Adam. I thought about the same, sort of, to actually do the analysis. It seems that it's there, but we haven't finished it. So it needs to be shown whether if we predict the values, then we sort of measure the same thing.
Mm-hmm. Yeah, lots more to do. Good. Andrew, you've got your hand up.
Yeah. So Joanna, absolutely fantastic, just wonderful. Cutting edge work. So a big theme for us is reproducible analytical pipelines so that people don't have to climb the same mountain that you've climbed. So can I get a sense of how easy is it to can some of these analytical approaches for further use? And secondly, are you happy you've got a conduit to make them available as we build this community?
Yes, I think that's very important, and we have it. We try to integrate it within all our work. So we have sort of GitHub pages that are associated with this manuscript to be able to provide, the workflow and the code for this analysis to be reproducible. But especially for metabolomics, if we talk about untargeted metabolomic data, it varies so much on the equipment and the parameters that are used on the equipment that it may be hard, even if they have the same sort of code or parameters.
Thanks.
And that's why we need humans and not ChatGPT, I should say.
True.
We're not redundant just yet. No.
We're not quite redundant.
Good. If there are no more questions for Joanna, I'll say thanks again. Do pop them in the chat if they spring to you later. And we're going to turn our attention from the metabolomics to the work on proteomics. So I think we've got a double act from Arun Hingarani from UCL and Mike Pietzner, who's in Cambridge and Berlin.
Yes. Hi. I think Mike's going to kick off.
Great. Are you in the UK or Germany today, Mike?
I'm today in London, so
London. Okay, very good. Good to hear where people are sitting.
Yes. I hope you can see the slides.
Yeah, that's great.
So as Adam already alluded to, so let's do some proteomic work. And I mean, why we're interested in proteomics fairly obvious, because it sits right at the center of how biological information flows. So we have our genome, where all the blueprint sits, and this is then somehow translated into messenger RNAs and these kinds of things. But then actually the proteins are the one physical or molecular entities who do all the work. They either facilitate metabolism or directly infer with the with our health, and they enable crosstalk among organs, and most importantly also they are the majority of drug targets. So that's why we have a specific focus, and it's what we specifically focus on are what we call PQTL. So these are single base pair exchanges, so one letter exchange in the genetic code, which may have a fairly drastic effect on how much protein we can measure in plasma. And this is really a real life example here. So whether you have a G or two Gs in your genome, or a G and a C or two Cs, it makes a massive difference how much protein we can measure in your blood. And I mean, we haven't been the first. There have been multiple efforts looking into these. And apparently very recently, with now UK Biobank also coming along, and they provided massive amounts of these PQTLs. But the question is, why do we need more, or what do we do to understand better? And we've been doing quite a lot of research on how do these different platforms there are to measure these proteins at scale, namely SomaLogic, which uses fancy little DNA fragments which really neatly fit in their three-dimensional conformation to proteins, compared to what is possibly the more traditional way to measure protein abundances, using these antibodies that bind to the protein, and then we have some kind of readout to see how much protein there is. And when we compared those methods from this large renders, we saw quite a few interesting things. First of all, we see what we expect. So if we look at, for instance, leptin, the fat hormone, it correlates really well. So when we look at the SomaScan values and compare them to the Olink assay, it aligns really nicely. However, there is also things where they don't agree at all. And even weirdly, there are even things where SomaScan says, okay, this is a high protein value, but Olink will tell you, no, it isn't. So it's an inverse correlation. And this basically means every inference we draw based on the measurements we very much depends on the measurements we do. So that's why we embarked again on a proteogenomic study, and this is work led by me and one of our stellar PhD students, where we used one of the most recent proteomic technologies from Olink called Explore and Expand, and measured those proteins in the Epic Norfolk study, and then again combined this with the genetic information to then go and see, does this inform us actually when we have these PQTLs? Are they disease relevant, or can we identify disease relevant proteins? And this is what is also depicted here on the right hand side, where we have identified a protein called gastrin releasing peptide, which seems to be related how thin or thick we are, which then translates into our higher or lower risk for type 2 diabetes. And in total, the discovery was only like 1,000 people. I appreciate this is a very small number for GWASs or genetic studies, even now in the field of proteomics. So it was even more staggering to us that we still saw novel things that haven't been seen in like studies 30 times the size of ours. And we did some fancy statistics to get a better idea of particularly those genetic variants that sit close to the protein encoding gene. And this is what I just mentioned. So each dot here would represent a genetic variant in the genome. And the higher the dot sits in this plot, the more confident are we that this SNP associates with the protein level. And you see on top, actually, that we identified more than 200 signals that haven't been reported. And again, this is in a very small study, actually, but using a novel technology. And then you can immediately ask, okay, does this make any sense? Is this somewhat relevant what we see because it's new, it's not really, doesn't replicate with other platforms? And this is where we then start integrating phenotypic data, because we do believe that when we start stacking up, how does the genetic signal look for the protein? And does it align with a genetic signal for a disease? Then we are much more confident that we are actually looking at biology and not some technical artifacts, for instance. And when we did this at scale, we can start drawing these networks of protein disease dependencies. And so basically what you see here is each of these white rectangles, put a laser pointer here, each of these white rectangles would be a protein encoding locus. And this is then linked to a couple of often diseases. So proteins are fairly, have pleiotropic effects. And you see this in the color gradient for some of those proteins that really have an effect on a whole array of diseases, which are very diverse and often treated by different clinical specialties. And it's really important to understand these bits. But coming back to a more tangible example, so what we identified is, put it an interesting, possibly even drug-relevant example is gastrin-releasing peptide. This is a hormone which stimulates the secretion of gastric acid, but it may have also other functions which haven't been really elucidated yet. And I think this is one of the great things when we start integrating these different layers of data, that we can identify proteins that we have been known for quite some time, but never really got to the bottom what they actually do, and apart from some obvious things. And what you see on the left side here is basically, okay, this is now zooming into the genomic region, and each dot here would be a genetic variant. And you see there's a couple of genetic variants that strongly associate with the protein, and they also strongly associate with different markers or measures of total body fat or body mass index. So, and this is in an inverse direction. So if the level of the protein is higher, possibly even lifelong higher, because you have this genetic variant, then it seems to be that these very same people are also thinner, and then most likely develop diabetes, don't develop diabetes. And this actually really nicely aligns with trials in humans, also some mouse studies, where when you give this protein to people as a human recombinant protein, it reduces food intake and leads to weight loss. So that's why we think it's a really promising area to pursue, because it's also very much mimics what is currently very effective as new obesity agents that also potentially protect people from transitioning from obese to type 2 pre-diabetic or diabetic conditions. And so this is called multi-omics. One thing that has been a massive topic recently due to data availability in UK Biobank is gene burden analysis. So when we look at very rare genetic variants which have a massive effect, so basically mutations more or less. And so we set up to really compare what can we actually get from these two very different resources of data. So for these roughly 3,000 protein coding genes, which we can cover by the proteomics technology, how does this compare if we do this exercise using these rare variants that have now become available in UK Biobank? And what we actually eventually see is that these two sources are very complementary. So the information where we link proteins to diseases there's only small overlap with what links to when we have loss of function mutations in those genes. So that's why I think it's really important to get multiple omics resources into the game, because they all can point you to something very different. And one thing that, when you're lucky, it converges, and then it establishes these really nice dose dependence relationships. So again, looking at the locus, so this protein TIMD4 nicely co-localizes, as we call it, with measures of lipoprotein metabolism, like LDL cholesterol. So more of the protein seems to reduce your LDL cholesterol levels. And the thing is, if you see this from the protein perspective, you're in the common end. So this is a common variation we see in the genome. There's a small effect on these blood lipids, but when we go through more severe effects, so where the protein is no longer functional, then we have even stronger effects on those lipid levels. So this is then where we can also think of establishing these dose-response relationships, which are really informative if we want to develop drug targets. And Mina is currently also pushing forward to get larger sample sizes. for these technologies by pulling together a diverse set of cohorts, more than 40 across the world, which bring together different bits and pieces of data for those proteins, because historically the vendor Olink distributed those proteins on different panels. So you have measured 90 proteins at once, and some cohorts have these 90s, other proteins have other 90 proteins, and it has really gone through a very tedious exercise putting all this together to be able then to really investigate what we are interested in the multi-omics consortium, to understand what actually drives differences we see across cohorts, and how does this impact all the inference we draw from. And then now we're then able to compare this to a very large set where everything is like same sample type processing, very homogeneous cohort, and you can Malbank to understand these bits and pieces better. And with that, I think I hand over to Arun.
Thank you, Mike. As Mike mentioned right at the beginning, not only are proteins the main effector molecules in biology, they're also the targets of most drugs. And so I just wanted to give two examples of how we've been integrating genomic and proteomic data together to answer important therapeutic questions. The first example shown in this slide is trying to identify new therapeutic targets for heart failure using proteomic data and Mendelian randomization. And this is work that's been led by Albert Henry and Tom Lumbers at UCL. Tom Lumbers was an HDR UK Rutherford Fellow. And this is working with the Hermes and Scallop Consortia using the Olink proteomics platform that Mike mentioned. So a number of proteins were found in observational studies to be associated with risk of heart failure. But of course, in observational studies, those associations may not be causal. However, by linking genetic with proteomic data using the pQTL approach that Mike described and applying Mendelian randomization, it's possible to differentiate those associations that are causal from those that are due to confounding. And when Albert and Tom did this, they were able to hone in on 8 proteins that had strong evidence for a causal association. And many of these were also potentially druggable. One protein that stood out was adrenomedullin. This is already a target of interest in early phase drug development for heart failure. And so by providing genetic support for the target, it provides increased confidence to take this target through into later stages of drug development. Next slide, please, Mike. Thanks. And then this is another example focusing on a very specific drug target, the cholesterol ester transfer protein. This has been of some interest as a drug target in cardiovascular disease for some time, but a range of drugs that have been evaluated in phase 3 clinical trials listed in the columns on the right-hand panel, torcetrapib, dalcetrapib, and evacetrapib, were considered to have failed in preventing coronary heart disease in these trials. And then a big question is whether this was a failure of the target or a failure of the compound used to modify the target. One way of gaining insight into that, using proteomic data on circulating CTP and integrating genetics, is to compare the profile on a range of biomarkers and disease endpoints using genomics, which should provide a clean readout of the effects of a specific potent drug with no off-target effects, versus the effects of the compounds that were actually used in the clinical trials on those same endpoints. And what the panel on the right illustrates is that torcetrapib and dalcetrapib don't reproduce that clean genetic effect that you see with the genetics in the right-hand column. Anacetrapib and evacetrapib are both much closer to that. And in fact, anacetrapib was shown in a very large phase 3 clinical trial to be effective in heart disease reduction, although the manufacturer Merck chose for financial reasons not to take it forward to licensing. So we've been doing some work with another pharmaceutical company that's taking another CTP inhibitor forward and shown that the target looks like it's valid, looking at the left panel now, to reduce the risk of coronary heart disease, but also chronic kidney disease and heart failure, but that there might be an adverse signal for age-related macular disease, illustrating how this type of approach can be used both to identify efficacy and safety signals for a target and therefore inform the design of future trials and whether to prioritize a particular target in a particular disease. So two examples here of how using genomic and proteomic data together can help gain insight into important therapeutic questions. This is an area of growing interest, not just in academia, but also in the pharmaceutical sector. So thanks, back to you, Mike.
I mean, that's basically what we should always emphasize. This is a large body of people who contribute to all this work and who led it. So I think it's always nice to acknowledge them at least somehow, only we being the ones having the privilege to present.
Quite right.
Fantastic, great duo, some incredible examples in there. And I think, you know, if you needed a 15-minute argument of why I think proteomics and genomics is useful, then I think that captured many of the examples that one would want to prove that. So brilliant work, and thanks for condensing it into a short pitch. John, you had your hand up first, I think.
Thank you. Thanks, Mike and Arun, super stuff. Could you comment, most of the examples you gave were based on binding assays, Olink, SomaLogic. Could you comment on what you think going forward is the priority for mass spectrometry-based assays and what potential added value or not that could provide?
Mike, do you want to take that?
Yeah, I can certainly take that. I mean, when we talk about plasma, this is a major challenge for mass spec proteomics because you have a I think abundance difference of 10 orders of magnitude, which isn't you can't capture this with mass spectrometry. So that's why these affinity assays are way better in getting to the low abundant targets, which are obviously the ones we're most interested in. So, but having said this, there's a set of proteins that are better, fairly abundant and well targeted by mass spectrometry, which then also allows us to dive into some of the discrepancy we see where we see different readouts of these affinity-based assays, but then actually mass spec can be very telling to understand which isoform of the protein. So just because we call it a protein doesn't mean it's always the same. So it differs by different post-translational modifications and all these bits and pieces. And this is where I think mass spectrometry can really help. But for this, it needs more sophisticated pre-processing steps in terms of how can we filter down the proteins we are interested before we ingest them into the mass spec.
Thanks, Mike. Juan Antonio, you are next, I think.
Yeah, hello. So yeah, I'm joining today just to learn about what's going on. I haven't been involved on this at all. So I'm leading the proteomics thing at the EBI. So I mean, I actually was going to make the same question at John. So I'm not going to repeat the question, but maybe I can also give my answer to that as well. So I mean, so of course, mass spectrometry is more difficult to because to multiplex is true, but now with I think that with the DIA techniques you know you get much better measurements in plasma, and also you get many more proteins that are not represented in Olink assays. And on the other hand, also the technology in a way is more established. And of course, the other aspect is what you mentioned as well, related to proteoforms. And so yeah, so that at least I wanted to give my point of view there. It is more difficult to multiplex and more technologically complex. So are there any concrete plans in the future for this consortium to move into that direction, or the immediate plans are to continue with you know, Olink and SomaLogic assays? Or are there any concrete plans in the future, or not really? That's also part of my question here.
Yeah, I think as a group we aim to bring together data where it exists across multiple cohorts, and so the most widely used ones you've heard about today, the SomaLogic and the Olink, but also to explore the opportunities, you know, typically starting with individual cohorts. And so some of the cohorts within the consortium are already, I think, trying some of the plasma mass spec approaches that are most scalable in terms of running at kind of thousands of participants, and UK Biobank, for example, is now piloting, I think, in 2,000 or 3,000 people several different approaches, because there's a lot of different competing or complementary assays in the mass spec place. So I think as a consortium, we will try and aggregate data where we can across studies, but it's good for us to embrace the new technologies as well within the cohorts and see where we can learn. And of course, there's people within the consortium with interests even deeper beyond that in the phosphoproteomic space or other post-translational modifications. So we've kind of started by trying to aggregate existing data, but I think as we move forward, thinking about generating new data and ways to achieve that as a consortium will be really important.
Okay, thank you, and congratulations everyone for the great work. I forgot to say that.
Well, thank you for joining us, and glad that we piqued your interest enough to join today to hear about the project. Great. Well, thank you very much, Mike and Arun, tour de force on proteomics. And so finally, I'm going to hand over to Mike, who's going to talk a bit about the final work package of the project, which was thinking about multiple domains of omics, not just in isolation. So over to you, Mike.
Great. Thanks, Adam. Hopefully my screen is showing up now.
It's coming. It started screen sharing. So yeah, here we go. It's in presenter mode at the moment.
Okay, this should do it.
Yeah, that's
good.
the job. Yeah, very good.
All right, great. So thanks, everyone. It's a pleasure to be presenting to you, on behalf of my co-lead, Professor Chris Yao, and on behalf of our whole work package. So by now, it's probably clear that multi-omics data is complex, but in integrating multi-omics data across cohorts, there's an additional challenge in that cohorts are a patchwork, albeit a beautiful patchwork of multi-omics data. There are different omes, that is, some have proteomics, some don't, for example. There are different platforms. For example, some have proteomics data on the Olink platform, some on SomaLogic, as we've just heard. And some even have different tissues. So most obviously, many have blood, but some cohorts may have other tissues like adipose. And the one constant, though, is that all or nearly all have genome-wide genotyping data, which is obviously special in that the germline genome doesn't change over time, unlike nearly all of the other molecular systems we're interested in looking at. Genotypes are therefore potentially very useful for multi-omic data integration. So here, I'll present a couple of papers arising from our work package of the HDR UK Multi-Omics Consortium, and each will seek to address a key question. So one, can we create a resource which allows us to impute multi-omics data using genotypes? And two, can we integrate omics data using an overarching biological model, for example, of a reaction network? Our most recent work, currently in press at Nature, focuses on using genetic variants to predict or impute multi-omic traits. And so this isn't a completely novel idea. It builds on others, of course, such as the fantastic PredictScan work of Eric Gamazon, Hakim, and many others. So here, our aim is to enable any cohort with genotype data to look at downstream biology by creating their own genetically predicted proteomic, metabolomic, and transcriptomic data. With this, they can perform a wide variety of subsequent analyses, for example, biomarker scans, as well as perform integrative multi-cohort analyses. It's of course not as powerful as generating one's own multi-omic data, but it is less expensive, obviously, and the difference in statistical power is proportional to prediction noise. So, there's still quite a lot to be gained when these predictors are applied to large biobanks like UK Biobank, all of us, et cetera. So importantly, it also enables multi-omic analyses for researchers who may not have the resources at their disposal to generate multi-omics data at scale. And that's really important, particularly in low income settings, LMICs, et cetera. So to build genetic scores for multi-omic traits that are used for prediction, we use the Cambridge-led interval blood donor cohort in the first instance. Interval has a wide range of multi-omics platforms available, including SomaLogic, SomaScan proteomics, Olink proteomics, Metabolon HD4, Metabolomics Nightingale Health, Metabolomics, and Illumina whole blood RNA sequencing. So this work was led by Yushu, a postdoc in machine learning, and involves the hard work of various collaborators many of whom are from the HDR UK Multiomics Consortium, led by Adam. So we used Bayesian ridge regression in 5-fold cross-validation in interval to construct genetic scores for each molecular trait. And for feature selection, we did some initial testing with a range of different variant set, but found that genome-wide significant cis and trans QTLs offered the best prediction R-squared with minimal overfitting. And to assess these genetic scores, we performed both internal validation in a 20% test set of interval, as well as external validation by engaging a global network of collaborators with diverse cohorts. So our external validations included those from, say, Fenlands from the east of England, Orcades and Viking from Scotland, UK Biobank, Northern Sweden Population Health Study, African Americans from the Jackson Heart Study, and the Singapore multi-ethnic cohort, which comprised the major ethnic groups in Singapore, Chinese, Malay, and Indians. And all of these cohorts, brought different combinations of the 5 omics platforms that I've mentioned, and which we've constructed genetic scores for. So here's a high-level breakdown. Overall, we were able to construct genetic scores for about 17,000 multi-omic traits, of which about 10,000 reached Bonferroni significance when predicting their directly measured trait in internal validation. And nearly 6,000 scores could explain at least 10% of trait variation, and about 400 could explain a majority, 50% or more, of the trait variation. And as expected, for all multi-omic platforms, there was a heavily right-skewed distribution of R-squared values, with a large number of traits being not or somewhat genetically predictable, and a small number being highly predictable. And interestingly, we found that some platforms were particularly amenable to genetic prediction, for example, Olink, while others, such as Nightingale, were less so. Oops. Sorry. So reassuringly, we found strong correlations between the R-squareds for interval validation and external validation across all platforms, indicating that overfitting was controlled and thus supporting the robustness of our models. However, this was all for data of European ancestries. And we found, as expected from complex trait analyses, that there was an attenuation, but not ablation of genetic score performance when we looked across ancestries. In this case, Chinese, Indian, and Malay from the multi-ethnic cohort in Singapore, and the African-American ancestries comprising the Jackson Heart Study. And from what we could tell, this was largely due to allele frequency differences and data missingness, but also it's also likely down to differences in linkage disequilibrium and perhaps some allelic heterogeneity as well. What was encouraging was that it allowed us to identify, at least in the Nightingale and SomaLogic platforms, which genetic scores could robustly transfer across ancestries. To demonstrate the applications of these genetic scores, we created a genetically predicted, i.e. synthetic, multi-omic data set for the 500,000 participants of UK Biobank, then performed a phenome-wide association scan for their transcriptome, metabolomes, and proteomes. And we identified over 18,000 associations at a false discovery rate of 5% across over 1,000 V codes, that is phenotypes. Our hits included new and known biomarkers, for example, LDL and IL-6 for coronary artery disease. And in this synthetic multi-omic UK Biobank data, we also validated previous associations of disease polygenic scores to protein mediators, such as those from a previous paper of ours from Scott Ritchie and co. From 2021. So we ended up validating 8 of the 11 putative causal mediators for coronary artery disease as well, which was pretty exciting. So integrating genetic scores across omic platforms yielded several examples of convergence on biological pathways and some pretty intriguing disease associations. For example, here the genetic scores from SomaScan, Olink, and RNA-seq, highlighting the association between JAK-STAT signaling and coronary atherosclerosis, which subsequently expands the picture when it comes to the well-known effects of IL-6 on CAD risk. So FAIR, that is findable, accessible, interoperable, and reusable data principles are at the core of our lab. So of course, we're keen to generate open resources for the wider research community. So here we developed the omicspread portal, www.omicspred.org, where we provide all the multi-omic genetic scores to the community for free and open validation, and for them to apply in their own research programs. So in the future, we're planning to vastly expand omicspread with the support of Health Data Research UK. And that's in terms of genetic scores, demographic diversity, and omic platforms. So do watch this space. I'll just keep going just as quickly as I can because I know we're behind time. So that was kind of telling you a little bit about how we can learn useful models directly from omics data. And this is speaking to my second question, what about leveraging our existing knowledge about biology? So here it gets even more complicated, given the incredible, tangled complexity, indeed hairball-like nature of biochemical reactions. So for example, here's a snippet of human metabolism put together by the giant biopharma company, Roche. So we set out to not only impute omics data, in this case, transcriptomic data using genotypes, but also to map these genes to genome scale metabolic models, such as human 1. The advantage of this is that we're no longer looking at just concentrations of metabolites, but given their encoded stoichiometry, genome scale metabolic models allow us to estimate specific biochemical reaction fluxes, that is, enzymatic activity. So this is an important step in understanding, what the effect of a genetic variant or a set of genetic variants are on the downstream biology. So this is all the work of Carlos Foguet, a HDR UK funded postdoc in a lab, who put together an algorithm to estimate genetically personalized and tissue-specific metabolic reaction fluxes for all individuals in interval and the UK Biobank. We then used these model-based reaction fluxes to perform a fluxome-wide association study for coronary artery disease in UK Biobank. So overall, we were able to estimate 763 fluxes from metabolic reactions, and detected about 4,300 or so associations across the 229 blood metabolites that we could assess from Nightingale and Metabolon, at about a 5% false discovery rate. So we found that most reactions were functionally parts of lipid metabolism, which is consistent with a large number of associations with lipid metabolic features. And it was very reassuring that there was widespread consistency between biochemical pathways and blood metabolites. So for example, in liver, reactions of glyceride and phospholipid metabolism and of bile acid metabolism were enriched. And similarly, reactions from nucleotide metabolism and amino acid metabolism were primarily associated with the blood metabolic features of nucleotides and amino acids. So the fluxome association analysis also uncovered intriguing hypotheses which extend existing metabolic pathways. So for example, a major determinant of triacylglycerols, TAG, free cholesterol and cholesterol esters in lipoproteins was a sequence of reactions which we termed the TAG to cholesterol esterification, or TAG-CE pathway. And our analyses revealed that the TAG-CE pathway flux was associated with the sizes and compositions of LDL and HDL cholesterol, aka the bad and good cholesterols, which are associated with heart disease, et cetera. So in associating fluxes with future disease, in this case, coronary artery disease, we found 92 significant flux to disease associations at a false discovery rate of 5%. And there are a lot of results to sift through here, but one I'd highlight is that one of our strongest signals was for the flux of histamine synthesis through histidine decarboxylase, which was shown to be associated with CAD in adipose tissue. So many of you are probably familiar with histamine as a pro-inflammatory mediator, and it's synthesized from histidine primarily in mast cells, which reside in most tissues, including the liver and adipose. And histamine has been also reported previously to be associated with atherosclerosis via blood lipids and lipoprotein fractions, as well as of course promoting inflammation. And indeed, it's a bit of a blast from the past for me, because one of the studies for my PhD found that whole blood gene expression of histidine decarboxylase was the top signal for various blood lipids and lipoproteins in both the Dilgom and the young Finn studies. So pretty cool to be reminded a bit of research which I did 15 years ago. So just real quickly, some acknowledgements. This is a broad acknowledgement slide for everyone who contributes, however minutely to these studies. But in particular, as I've shown, the real stars here are Yushu and Carles, and of course my co-lead Chris Yao. So thanks very much. Happy to answer any questions.
Wonderful, thanks, Mike. Great to see some of the innovative things that you and your team have done with the data, going beyond individual platforms or individual measurements, and great to see the open deposition, the catalogs, and the ability to download all the information, the scores. Where do you think you might take this next, Mike? I'm interested to hear what you think the future is in this domain of work.
Yeah, so absolutely. So I think there are a few main dimensions here to extend. So one is obviously, and we tried to do this as much as we could in the omics paper, is looking at demographic diversity, not just in terms of looking across ancestry, stroke, ethnicity, but also I think it's important to start looking across male, female, so biological sex at least, and also across I would hope to, indices of deprivation, socioeconomic status, et cetera. So I think that's one extension, which would be really good to see early on as this field of genetic prediction of multi-omics data develops, so that we get ahead of the game and we don't end up in a situation like we are with polygenic scores of more complex diseases and phenotypes, where we have relatively good performance in white Europeans, but quite attenuated performances as we get further and further away, and in some cases a belated performance when we go to individuals of African descent, and in particular those who are in Africa. So yeah, I think heading that off at the pass at this stage with molecular data is one of the priorities. And one thing the other one I'd also highlight is increasing both sample size and the platforms that we have generated genetic predictors for, which facilitate the downstream data integration, hopefully fairly clearly. So I think those are like the main areas of progress.
Thanks, Mike. Yeah, certainly the diversity piece, you nicely illustrated the challenges there, and the initial attempts to try and see how some of the tools that we generate in people of European ancestries will translate across the world, or to people of different ancestries in the UK indeed. So yeah, that's going to be a big challenge for the next 5-year cycle. And any other questions for Mike? I had a joint question perhaps for you and Joanna, if Joanna's still on, which is we saw attempts in both your presentations of trying to impute data or kind of predict it from some information. So Mike, you were basing it around genomic data and trying to predict molecular traits. And Joanna, you were, I think, using other metabolomics platforms. Where do you both see the relevance of other information that might be collected in the cohorts, and that might be basic information like age and sex, which presumably has some reasonable influence on imputing these, but I guess one could even then think about, for example, health data that we have from the linked medical records or other domains of information rather than just genetics or metabolomics. Joanna, do you want to comment on that one first?
Yes, I think definitely we can, and we are using things like age, sex to increase the predictive ability. But as you say, even more, we can think of health data as well to make those predictions. We haven't tried that yet.
Yeah, and totally agree. So one of the next steps that we're looking at is incorporating age and sex alongside the genetic scores for prediction, and looking at sex-specific trajectories of these molecules. So yeah, the only, in terms of in using other data, as you sort of increase the number of features that you use to predict, you do have to be careful of some of the biases that might be incorporated, particularly when you start looking at the patchwork of data that's in electronic health records, for example. And at least in our space, you're looking at enrichment of biological pathways is also something which can be affected by that bias. So as you expand the complexity of these models, you know, some of the little traps get laid in front of you. So we're just trying to unpick that at this stage. Yeah.
Thanks. It seems like there's plenty of opportunities going forwards. So I don't see any other hands raised or questions. So thanks to everyone who presented in the second session. It was really incredible showcase of you know, some really excellent world-leading research in these areas. We've been blessed with the quality of the people we have around the UK who've been able to come together on these you know, excellent data resources, but to really harness them and to really squeeze out insightful, meaningful results that will you know, ultimately lead to tools and changes in clinical practice and development of drugs, etc. So I hope people have enjoyed today's insights sharing day. Hopefully the challenges and successes that I kind of laid out at the start, you've seen answered to some extent. I think, as we talked about right from the beginning in the PPIE section, we're very keen to reflect, to learn, to see what we did well, what could still be improved. So I've posted in the chat some of the key email addresses if people do have comments. Particularly Amanda was very keen to, I think, collate feedback on the PPIE aspects. And we're still, I guess, as we've heard from some of the acronym comments or discussions in the chat, we're still trying to improve on doing mixed audience workshops. So yeah, do feel free to reach out to Amanda on that front. I think if you have comments about today's workshop or about the science and things, then I've posted my email address, and Loma Ramona, who played a huge part in organizing today's event as well. So we hope to continue to present our science as it continues in its new form in the next 5-year cycle of HDR UK. So we'll be interested to get your feedback on the format. So thank you to all the presenters, everybody who worked on all the papers and the great projects that we've put out today. And thank you for those of you who've attended, whether you've been closely involved in the project, new for the first time today, and particularly our public and patient representatives who've been really invaluable. And it's great that you've been able to join and hear about some of the work. So thanks very much.
Well done, Adam.
Thanks, Andrew. We'll draw it to a close there.